Three, two, one. We are live now, sir.
Good evening, everybody. Myself, Dr. Vishal Dave, Medical Advisor, Intas Pharmaceuticals. On behalf of Intas Pharmaceuticals, I welcome you all for this Synergy 15 webinar on the topic Unlocking Genetic Secrets Part 2. Before starting the webinar, we will have a prayer.
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः.
It is my privilege to introduce Dr. Monica Singh, the convener of this webinar. Dr. Monica Singh is President-elect of Bhopal Obstetric Gynec Society. She is Founder-President of ISOPAP chapter of Bhopal. She is National Executive Member of ISAR. She is also Joint Secretary cum Treasurer of MPIAG as well as Coordinator of Foxy Infinity Committee. Over to Dr. Monica Singh.
Thank you. Thank you so much, Dr. Vishal. And thank you, Intas, for giving us this academic platform for conducting the Synergy webinar series. Today, it's my proud privilege to announce that we are doing this webinar number 15, the episode number 15 on Unlocking Genetic Secrets Parts 2 in association with ISOPAP chapter of Bhopal, which has been newly formed last year, along with Sagar OBGY Society. So, because of our association with Foxy Societies, we are always fortunate to get ICOG points granted to our webinars. So this time we are having 4 ICOG points. A lot of fellows join our webinars for the purpose of academics and also as panelists and chairpersons. So today we will be further unlocking the genetic secrets, which we started doing on 14th of May. We had part 1 of this webinar, wherein we had two very good talks on the first trimester screening tests, as well as the validity and importance and interpretation of prenatal diagnosis by genetics. And today, because ye dil mange more, so we really want to know when we have gone the reverse way, actually. We have done the clinical part, and today we want to see what genetics is all about and how we can apply that knowledge of basics to unlocking genetic secrets. So this is part 2 of the webinar, and we are very, very proud to welcome amongst us our chief guest, Dr. Parul Kotadawala sir, the immediate past chairperson of ICOG, our guest of honor, Dr. Ashok Kumar, who is vice chair ICOG, and Dr. Sarita Bhalerao, who is secretary ICOG. Madam will be joining after a little while. And our special guest for the evening is none other than the charming Dr. Kanti Bansal, who is Director, Safal Fertility Foundation, Ahmedabad. So welcome to all our guests. I'll walk you through the program. We begin with the structure of DNA, the intricacies of which will be unraveled helically by Dr. Doyle Bose Pandey. And we have another very interesting talk coming up right after that on screening of hemoglobinopathies, because that is always a confusing thing, because we generally don't do it as routinely in our OPD practice as other tests. So this will give a lot of clarity to the subject by Dr. Jia Chawla. And thirdly, we have a very important talk on screening and prevention of chromosomal anomalies by Dr. Deepika Deka. So without wasting any more time, I would like to extend a very warm welcome to our chief guest for the evening, Dr. Parul Kotadawala sir. Sir is immediate past chairperson of the, the luminous, the illustrious body of gynecologists, that is ICOG. He is a renowned endoscopic surgeon and consultant gynecologist, professor and head of Department of OBGY at Sal Institute of Medical Sciences, Ahmedabad, and the Kotadawala Women's Clinic at Ahmedabad. He is also senior consultant at Zydus Hospital, Ahmedabad. Sir holds the post of immediate past chairperson, ICOG. He also has been the vice president of ISOPAP in 21 to 22, chairman of the education committee of CEFOC. The slide is blurred, Dr. Vishal. The India coordinator of NESA, that is the New European Surgical Academy, India coordinator of IAS, that is the International Adhesion Society, vice president of FOXY in the year 2006, president of the state organization of OBGY of Gujarat, that is SOGOG, in 2009, and the president of Ahmedabad Obstetric Society in 2007 and 8. Sir has been the chairperson scientific committee of the widely acclaimed AICOG, which was held in 2017. We are so honored to have you amongst us, Arul sir, and we would be blessed to have a few words of wisdom from you.
Hello, I hope I am audible.
Yes, sir.
Thank you so much, Dr. Monica. It is indeed very unusual for me to be a chief guest. Generally, I feel that a person who retires is the suitable person for chief guest. I, in my heart, do not feel that I am yet retired. Although, yes, I am past chair of ICG. Thank you so much for asking me to be part of this very important webinar. Genetics has always been a very confusing subject for us. And right from my student days, I still remember that I used to find an option where I can avoid answering questions on genetics. So, but it is so very important in our practice. And with more and more knowledge on genetics, definitely we can do something for our patients. Previously, it was just that knowing the reason was the end point. Now we can intervene and do something for our patients. And all these three topics are superb. Structure of DNA, of course, will be a googly for all of us, but I am sure Dr. Is it Doyle? How do we pronounce her name?
Doyle.
Doyle.
She's a Bengali.
Okay. It would be interesting to listen to her. And then the other two are really important topics in our clinical practice, screening of hemoglobinopathies, especially the eastern part of Gujarat and northern part of Maharashtra, and I believe some western part of Madhya Pradesh have a belt, tribal belt, where we find so many cases with hemoglobinopathies. And of course, Dr. Jaya Chawla will address that, and Deepika Deka is a great clinician as well as a scientist for this. And screening and prevention of anomalies would be so very important. I also see here my colleagues, Dr. Ashok and Sarita. Sarita will join later, as you said. And is Kanti also there? I sort of welcome them all. And you have so many senior colleagues as chairpersons. And I am sure this webinar will be very beneficial to all colleagues. And in the morning, as we discussed, ISOPARP is a platform which is the right platform for interaction between specialties as well, perinatology. And so you have very rightly chosen this topic for today's webinar. Let us have the presentations just now. Thank you so much again for asking me to be part of this.
Thank you. We are delighted to have you amongst us, sir. And we would be really happy if you could, enjoy the talks as well. The speakers will be.
I'll stay over as long as possible.
Thank you. Thank you, sir. And I think Dr. Purva, Shweta, would you like to tell me when Ashok sir joins, or has he joined?
No, sir, it's not your.
Yeah, so we will be introducing our guests of honor as and when they join. We have amongst us our guests of honor, Dr. Ashok Kumar sir, who is the Vice Chair ICOG, and Dr. Sarita Bhalerao ma'am, who is Secretary of ICOG. As soon as they join, we'll do the honors. And today we have amongst us our special guest, Dr. Kanti Bansal ma'am from Ahmedabad. Madam is Medical Director of Reproside Biosciences, Bengaluru, as well as Director of the Safal Fertility Foundation, Ahmedabad. She is a pioneer in the field of ART, not only in Gujarat, but whole of India. And many know her as mentor and teacher, and always lending a helping hand to all students. She is a pioneer in introducing AI in her lab, virtual reality and haptics in ART simulators. We attended a very interactive webinar wherein we talked a lot about AI and virtual reality, how it can be introduced in ART. Madam has been awarded RCOG Distinguished Service Medal in 2020 She has special interest in PCOS, endometriosis, and women's healthcare, has published 12 books on PCOS, endometriosis, infertility, ART. These books are referred to very fondly by all medical students and practicing clinicians. She's also a PhD guide at Gujarat University and has trained more than 2,000 gynecs in this field. We welcome you, Kanti ma'am, and we would like to have a few words from you, ma'am.
Yeah, thank you so much, Dr. Monica. First of all, I congratulate the entire team, including Vishal Dave. You know, those people are very important for us who host these things and encourage us to do. Otherwise, it's not possible for us as, clinicians. And I would really like to salute one couple in our fraternity, that is Dr. Randir Singh and Monica Singh. I've seen them when they're so, I will call you small. And I used to be so impressed that they are presenting year after year in the ISHRE conferences, such nice, scientific topics. Very proud of you both. So enthusiastic, and, most impressing is you people are academic, at the same time not grumpy, all the time smiling. I really admire, truly appreciate. And what a fantastic topic you have chosen, Monica. I know about the the past one, and in continuation, you have one more. But I'll tell you, you'll have to have one more on my request. I'll give you the topic also. And unlocking genetic secrets is a fantastic topic. I don't know, so many secrets are going to be revealed today by all the three fantabulous speakers. I know Deepika personally, and the other two also. I just saw Viol. All these people from Calcutta, I tell you, they have such brains. We can't compete with you all. With the rasgulla in your mouth, you'll talk, but then that's on a lighter side. And then to continue, I think in our gynec practice, we have a few of these disorders which have a genetic basis. Like, for instance, PCOS, it does have a genetic basis. We have endometriosis, which has, genetic basis, fibroids. Last but not the least, our own, Monica's and my branch, infertility. In that we have premature ovarian insufficiency, which also is having this genetic basis. And also there are other groups as well which do have to we have to deal with the genetics. And then like reproductive genetics is a big branch. And then there is another branch which is the cancer genetics. That is another one very big group. So we have to deal with all these things. And and there are so many emerging gynecologic tools and technologies. And so I think you people are today going to do the decoding of the genetics. And I would request Monica and Randir to get, and the ISOPAP of Sagar and Bhopal to get one more for all the other things that I mentioned. And there are a lot of newer things like, whole genome sequencing, CRISPR and gene editing. And we also have polygenic risk shares, etc. So these are some of the things I wanted to tell you. So I would particularly request Monica to have something on the PCOS and the endometriosis genetic basis. You need to have some more, webinars. Yeah, so I'm wishing all of you great success in these webinars, and thanks once again and congratulations. Yeah.
Thank you so much, madam, for all those wise instructions and suggestions. And we'll definitely keep it in mind that genetics cannot be separated from any of the topics, in fact. Whether
Yes.
we deal with any pathology, gynec pathology, infertility pathology, we have to delve into genetics, and we'll definitely have more sequely to this. And thank you so much, madam, for joining and always guiding. And now I would like to thank my co-convener for today, Dr. Randir Singh. Madam has already talked about him. He has a very big support system to all our academic and clinical activities. He's very active in ISAR. And I also have with me my 5 beautiful coordinators who you have interacted with in the last 2 weeks. They have been, selecting faculties, going through the list, calling them, and giving them sessions, taking their permissions. So we have Dr. Purva Bhatkur, who is assistant professor at GMC Bhopal, Dr. Nandini Singh Bhati, who is assistant professor again at GMC Bhopal, Dr. Sweta Agarwal, who is director at Ashoka IVF Care Bhopal, Dr. Richa Samadia, who is consultant reproductive medicine at Bansal Hospital Bhopal, and Dr. Shikha Gupta, who is director at Deep Shikha Hospital Bhopal. So they all are doing their bit very brilliantly, and they are all accomplished academicians, clinicians, very active girls. So soon they will be stepping into our shoes and doing programs even better than this. So now, without wasting any time, we move on to session 1. And for that, I hand over to Purva.
Thank you so much, ma'am, for your kind words. Now moving to our first academic session, that is on structure of DNA. I invite chairperson as Dr. K.S. Jayarani Kamraja, ma'am, Dr. Shabana Sultan, ma'am, and Dr. Jagriti K. Nagar, ma'am. Dr. K.S. Jayarani Kamraj, ma'am, madam is Associate Secretary, World Association of Sexual Health, immediate past president, Obs Gynae Society of South India. She is Vice President, Tapisar, Joint Secretary, TNFOG, Advisory Council Member, World Association of Sexual Health, Joint Organizing Secretary of very successful ISAR 2015, member of Board of Studies at Dr. MGR Medical University. Also, she is very active member in all the national organizations, and she has to her credit various awards like Best Doctor Award, Best Achievement Award, Service Award by EXNOR 2010, Mega TV Achiever Award. We welcome you, ma'am, to chair our session. Next, we have Dr. Shabana Sultan, madam. Madam is presently professor and head in my department, Department of Obstetric and Gynae, GMC Bhopal. She is a very active member of Bhopal Obstetric Society, and presently she is also vice president of Bhopal Obstetric Society and has held various executive positions in our society. She has participated and published various papers in national and international journals, and has been part of various international projects at Howard University. She has special interest in gynecology and high-risk pregnancy. She is master trainer of cervical cancer program. We welcome you, ma'am, to chair the session. Next, we have Dr. Jagriti K. Nagar, ma'am. Madam is immediate past president, Sagar OBGY Society, and also associate professor, Department of Bundelkhand Medical College, Sagar. We welcome you, ma'am. Now I hand over session to our chairperson to introduce our speaker, Ms. Doyle Bose Pandey.
Thank you, Purva. Thank you for the introduction. And I'm very greatly honored and privileged to be here as a chairperson for this session. And my special thanks for the very energetic couple, Monica and Randir, for making this Synergy Club meetings a very popular one. And it is having a lot of academic value, which is really greatly appreciable. And I really wonder how both of you, with a busy work schedule, to have such a nice academic program organized in a beautiful way with a beautiful lot of conveners and all the people. Really a very great appreciation to you, Monica, for arranging. Thank you so much for including me in this CME. And of course, this unblocking genetic secrets of part 2 of yours is really mind-blowing, because when you look into the clinical practice in genetics, many of us may not know the basics, but it is a coming and upcoming that we should know about what is the basis. So to start the basis, it's always good to start with the physiology, to know the basics, to revamp our past knowledge about the DNA. And I'm really happy to have here Dr. Doyle Bose Pandey. Madam is from, can I have Madam CV? Yeah. She's actually a certified senior clinical embryologist. She's the lab director at Indoor Infertility Clinic. She's the course director of the trainer in embryogeny and former product architect at CSC Technologies. And she has a special interest in analysis of blastocyst culture, genetics, and use of AI for IVF lab and automation. And she's the web designer and SEO specialist and avid blogger. And she's Madhya Pradesh ISAR Executive Committee member, life member of ACE, IFS, FPS, and ISAR. and she has contributed chapters on epigenetics in ART, troubleshooting in semen preparation in ART, and AI in ART. We welcome you, Dr. Doyle, to share your presentation on the structure of DNA, which is very important for all the clinicians to brush up their knowledge about the basis about the DNA. Over to you, Doyle, for your presentation on this.
Thank you, ma'am. I'm sharing my screen now. Is my screen visible?
Yes, yes,
Yeah,
Doyle.
yeah. Make it as a full screen.
Is this visible as a full screen now?
No?
Yes,
Yeah,
ma'am.
yeah, now it's okay. Yeah.
I
Not
think
yet.
it's a lag. Just confirm if it's visible now, then I'll start.
Yes, ma'am.
Okay. Let
Yes,
me
go ahead. It's visible, Dr. Doyle. Yeah.
All right, so I think I have a tough job at hand because Dr. Parul just mentioned that DNA and genetics is like a googly. So I'll try to make it a slower one and try to make it as simple as possible. I think my background in mathematics and electronics engineering makes it easier for me to understand genetics because genetics is almost like math. If you know the basics right, then concepts are not that difficult. So today what we will try to do is in a normal, regular paced fashion, we'll not try to cram up too much of information, otherwise it definitely will become a googly. We'll try to go from nucleotides to DNA to chromosome, genes, the central dogma, the very important concept, transcription, translation, and on that build on cytogenetics. If we have time enough, we will go to genetic mutations, but I think that we will have to stop at genes, but we'll see how we go forward. All right, so human genome, this you have seen plenty of times. You might find it boring at times that, comparing a human genome to an instruction manual. So this is the easiest analogy to understand what genes and DNA are. So if human genome is like an instruction manual, as you see on the screen, then the nucleotide base pairs, which are the nitrogenous bases of the letters A, C, T, and G, these are like the alphabets on your instruction manual. Now, when you arrange these alphabets in, in a meaningful sequence, then you create words. These words are nothing but segments of a DNA, which are called genes. These genes and this DNA is wound on a structural protein, which is called the histone protein. And then these wound protein is then consolidated and in a small space called the chromosome. So the chromosome becomes like a chapter in the book of your instruction manual. So I think it always makes sense to see some videos. So these are the 4 adenine, cytosine, guanine, and thymine nitrogenous base pairs. They are complementary base pairing in a step ladder-like fashion. And this is the double helical structure of a DNA. They are bound across these blue things called functional unit of a nucleosome. And these nucleosomes come together in a very compact structure to form a chromosome. So the purpose of the chromosome is to, compact the genetic information in as much compact possible fashion so that they can stay within the nucleus of our cell. So after looking into so I hope from a gene to a chromosome, it's clear that genes are segments of a DNA and the DNA in a compacted fashion is called a chromosome. Now, what is the function of a gene? It is very important to understand the central dogma of molecular biology, which says that one gene is coding for one polypeptide chain. So we will go slow with what do we mean by a polypeptide chain. So like we know, we have about 20 amino acids. These amino acids, when they are joined one after the other in a particular sequence, it becomes a polypeptide chain. And more than one polypeptide chains, they come together to fold as a protein. So basically, the function of a gene is to provide a genetic code that will allow some molecular machinery within the cell to form this polypeptide chain, and that eventually will go ahead and form a protein. So everything is a protein in our body. Everything is protein and enzymes. That is all that all these genes do. So now is the time to introduce you to the concept of transcription. This is a cells, like we have seen, the DNA, the helical structure of in a helical structure is present within the cell's nucleus. So what you see within the cell's nucleus, let me change the mouse pointer because I'm going to need it. I think the pointer is visible. So this is the cell's nucleus. This is the DNA, which is as a step, a double helical structure. So the job of transcription is to, if you just take it very simply, that the job of a gene is to provide a genetic code, which has to now assemble all the building blocks available and then make a protein out of it. So it is not going to do all of this within the cell's nucleus because you hardly have any space there. But you want the instructions to come out so that somebody can read those instructions and make the polypeptide chain, which will eventually fold to a protein. So how does the DNA provide this instruction to the cell's cytoplasm? So the easiest way is to copy this instruction into a messenger RNA, which is just one single strand like visible here. So a molecular machinery called RNA polymerase will sit on the DNA. It will, it's a complex procedure. I don't want to get into the details of how a fork is created and all that, but simply put, it is simply going to copy the genetic code, whatever it is reading on, say, one strand of the DNA. It will copy it, and then that messenger RNA is going to travel to the cell cytoplasm. So the job of the RNA polymerase is done. It has copied the genetic code from one gene, and it has now passed that information through the nuclear pore. These are nuclear pores, and it has come out in the cell cytoplasm. Within the cell cytoplasm, you have all the building blocks available as amino acids. Now, the job of the second molecular machinery, which is the ribosome, is to translate the messenger RNA. So what is translation? Translation is a process where you read the messenger RNA and you assemble the amino acids which are available one after the other and create a polypeptide chain. So what you see is this representation in the clip art is a polypeptide chain. More than one such polypeptide chains will eventually go on to form a complex protein. It will be folded as a complex protein. So the job of so that is the difference between a transcription and translation. Transcription is always done within the cell's nucleus, and translation is a process which will occur in the cell's cytoplasm with a final aim of converting the genetic code to a final folded, a polypeptide chain, which will eventually become a protein. Now, seeing the same thing in terms of, a double, this is the DNA structure. Now, how do you actually know which amino acid to join after which other amino acid? That is itself the genetic code. So genetic code is always presented in terms of codons. So a codon is a trinucleotide. Trinucleotide, 3 nucleotide. For example, ATG is a nucleotide, trinucleotide, it's a codon. CCT is a codon. So the molecular machinery in the cytoplasm reads the genetic code, 3 nucleotide base pairs at a time. It reads it 1 codon at a time. So this is during transcription, where the only difference is that the thymine is replaced by a uracil. Why is beyond our scope. Let us just like in mathematics, you learn tables and you don't go as to why. Let's not go to the why as of now, because it will get complicated as to why you need to replace a thymine with a uracil. Let's go with the flow. Thymine will be replaced with uracil during transcription. So what you see here is ATG is going to be converted to AUG because T is converted to a U. CCT, the T is replaced by a U. So while copying, you copy C as is, but the T is replaced by a U. The same goes with GAG. U copy is as is because there is no T there. So this is how each codon is read from the DNA during transcription, and it is copied on a RNA strand with just one replacement, where the thymine is replaced with uracil. Now, after this, when the messenger RNA is going out to the cytoplasm, we start the actual process of translation, where you will take an appropriate amino acid and you will join it to form a polypeptide chain. Now, how does an AUG, let us go back, how does this AUG mean that you have to take a methionine? And how does a CCU mean that you have to take a proline? This you have to see through a codon wheel. So just like logarithmic books and all, you have this coding chart called a codon wheel. It looks very complicated to begin with, but give it some time and it is not that complicated. So you always read a codon wheel from the inner wheel to the outer wheel. Okay. So what is this? This is nothing but telling what codon is coding for which amino acid. So if you have trinucleotide, it means 3 nucleotide, 3 positions, where each position can have one of the 4 letters. So by mathematics, it is 4 into 4 into 4 types of combinations and permutations possible. So 64 different possibilities of the 3-letter combination, where each position can hold 4 different values, A, C, U, and G, right? So that here you have 64 of these 64 different codons. 61 will code for 20 amino acid. We all know we have 20 amino acid in human beings, and 3 of them are used as stop signals. And what are those stop signals? UAA, UAG, and UGA. So if you look at the codon wheel, this is U, then you go to G, and this is A, and UGG. All of them, UGA is a stop signal, and UAA and UAG, these both are again stop signals. So 3 codon are reserved for stop signal, and AUG, which is methionine, is the start signal. A, U, and G. So this is how you read it. So AUG will always go for picking up a methionine amino acid. So this is the code for the amino acid, and this is the only, the start signal is unique. If you look at this entire wheel, you will see that, for example, leucine, which is an amino acid, has multiple ways of coding it. How? You have UUA, UUG. This is 2 of them. You again see that we will repeat it. This is again a repeat. So this is 4 different ways of coding the same amino acid. So of the 61 code, you can code 20 amino acids. So naturally, you're going to repeat more than one code for a particular amino acid. So it is very important to understand this codon wheel, because eventually, when you come to genetic mutations, just one change in a letter is going to give you a different amino acid altogether in the polypeptide chain. And that is the reason of presentation of a disease. So we will go back to our translation process. So we saw that AUG, according to the codon wheel, was methionine, which was a start signal. Start signal means that now you have to start assembling a polypeptide chain by the ribosome. The next one, you will see that a CCU, you can refer the codon wheel back and you will see that it translates to proline and so on. So this is how you keep on reading one codon at a time. You attach one amino acid with your increasing chain with a peptide bond. And this is why it is called a polypeptide chain. And this polypeptide chain, along with couple of other polypeptide chains together, combine to form a folded protein. So this is the entire purpose of the genetic code within your cells. Now, seeing the same thing using animation. So this is from DNA to protein. So this is your cell. Within the cell, you have a nucleus. Within the nucleus, you see the chromosome, which are tightly packed DNA structures on histone proteins, like we had discussed previously. And this is a segment on the DNA, which is a gene. Now, RNA polymerase is creating a fork and it is replicating the DNA to form a messenger RNA. So it is taking, reading the entire DNA and making a copy in terms of a messenger RNA, which is always single-stranded. Then there is a concept of splicing, after which you remove the non-coding part and then you transfer the entire messenger RNA to your cell's cytoplasm. Within the cell cytoplasm, another molecular machinery, which is your ribosome, it is now going to catch hold of the start signal within your within the messenger RNA, and then it is going to assemble one amino acid at a time by reading the codon and attach an appropriate amino acid at the right place, and eventually it will form a protein. So this is how we go from a DNA to a final product of a protein. So gene product is nothing but that polypeptide chain. So after covering the nucleotide, DNA, and chromosome, let's move to a bit about cytogenetics. So the word cytogenetics is a study of chromosome and molecular genetics is a study of gene. Like we saw, gene is just a small part on your chromosome. Cytogenetics is a studies the disease due to the abnormal number and structure of chromosome. So during cytogenetics, we are going to stick to chromosomes and whether they are normal in number, whether they are normal in structure or not. We are not going to go into genetic mutations, but in molecular genetics, we will go to studying diseases due to the mutation in the genes. And different techniques like karyotyping, chromosome staining, FISH, CGH, all of this will be used for studying cytogenetics. And for molecular genetics, you have different techniques like PCR, molecular cloning, DNA, RNA sequencing, etc. So these are just buzzwords when you hear what is cytogenetics. So cytogenetics is study of chromosome in short. Now coming to, after knowing the function of a gene and how it is, what is the content of a chromosome? So like we saw, DNA is only 40% of coding and non-coding genes. So within a gene also, you have coding genes and non-coding genes. So, but the DNA is not completely just coding and non-coding genes. So 60% of the DNA is packaging proteins. 48% of it is histones. Histones are those structure around which you are going to wind the DNA for a tighter, compact position. And you also have scaffolding and structural proteins, which form the part of a chromosome. Now, this I think it's very, when you read a karyotyping report, you always see a P and a Q, which is the short arm or the petite arm and a long arm. So a chromosome usually is not centrally divided into two segments. There are different presentations to a structure of a chromosome. We'll get to it. So in the center, you have a centromere. The ends of the chromosome are telomere. And what you see as the legs or the arms are the chromatids. Now coming to chromosomal states, the chromosomal states actually define how genes are regulated. By that, what I mean is, we all have 23 pair of chromosomes in all of the cells of our body, excluding our gametes, right? But we have different types of cell with different functionalities. If all the cells have same type of genetic code, why don't they function similarly? Is the main, this is mainly, taken care of by gene regulation. So we don't want all of the genes to be active all of the times. A skin cell has a different function. It should be coding for different kind of proteins. A hair is different, a liver cell is different. So gene regulation is the reason why, despite having the same number of chromosomes and same genetic code actually on all of the genes present in each cell of our body, why we have different types of cell which behave differently. So what do you mean by gene regulation? So for that, we have to understand a chromosomal state first. So chromosomal state, you have something called euchromatin. When you say euchromatin, it is where the nucleosome, which is the functional unit which is winding the DNA, are spaced apart. They will expose the part of the DNA where a gene is present, which is then active and can be coded can be transcribed to an appropriate messenger RNA. So here the DNA is accessible and the gene is active, and that is why it is called an euchromatin, because the letters are spaced away far. Now, at the same time, look at the other part, which is heterochromatin. It's very difficult to even read this, what I have written, heterochromatin. Why? Because the letters are very close by. It is very difficult to read without spacing these letters out. So these nucleosomes, when they come tightly close with each other, they don't have any segment of the gene where a molecular machinery can sit to copy that genetic code. So here the DNA is inaccessible and the gene is inactive. So these are two, euchromatin and heterochromatin are the two states of chromosome. And this is the reason why, this is how the genes are regulated. Taking it further, when you have a euchromatic band, then like we saw euchromatin, you have them spaced, the nucleosomes are spaced apart. When you have things spaced apart, light can pass through it. So naturally, even after staining, you are going to see lighter color through it in microscopy. So they will be lighter colored bands. So the lighter colored bands are the active regions of gene. At the same time, when you have a heterochromatin or a closed state, you have nucleosomes very close together with each other. It will obstruct light even after staining. And that dark region will be your inactive part. So when you read a karyotyping report, you definitely see a lot of light. You have a Gibbs staining where you have light and dark things visible. So that is how these the state of a chromosome impacts how it is visually seen under microscope. Now, types of chromosome in relation to the centromere location. So, like I mentioned, centromere is not always placed exactly at the center, but somewhat center is a metacentric position where the petite arm and the Q arm are almost similar, but not completely. At the submetacentric is where the centromere is at one, it shifts towards one side, making the petite arm shorter and the Q arm much longer. And acrocentric chromosomes, you have the centromere almost at the distal end, but not completely the end. So that is acrocentric. Telocentric is where you are completely at a distal end. But humans, we do not have telocentric chromosomes. We only have metacentric, submetacentric, and acrocentric chromosomes. So these are the type of chromosome based on merely the position of centromere and their physical appearance. So based on these positions, we have these 22 pair of autosomes and X and Y chromosome. Here, as you can see, when initially we tried to identify these chromosomes under microscope, they were ordered from 1 through 22 in order of their size, 1 being the largest and 22 being the shortest, but 21 is technically the shortest one, which depending on the number of base pairs it has. But I think microscopically it looked almost a little bit longer than 22. That's why it was numbered 21 first. So when we see a karyotype report, the chromosome 1, 2, 3, 4, and so on, they are numbered based on their size, chromosome 1 being the largest one and 22 being the shortest one, and XY chromosome. Now, types of chromosome, these are the chromosome numbers, and these are, so chromosome 1, 3, 16, 19, and 20 are metacentric. Chromosome 2, 4, 5, 6, 7, 8, 9, and so on are submetacentric, and chromosome 13, 14, 15, 21, 22, and Y are acrocentric. Now, why is it important to know which chromosome is what? This is almost like you have to memorize this, but what happens is when you actually see many karyotyping report, you will figure out that some of the errors like Robertsonian and all, they happen with ectocentric chromosome. You will remember that when you read the structural error. So it's important to just know what are the kinds, even if you don't remember the numbers, it is fine, as in which one is what, because it's not the job of the clinician to look at the chromosome and tell which one is what. Geneticists are there for that purpose. But it's good to know this. So karyotyping and gene staining. So karyotyping is nothing but a snapshot of chromosomes. The typical resolution is 5 to 10 megabases. When you go for high resolution karyotyping, it is 2 to 5 megabases. And karyotyping is used to detect a numerical error, structural error, large deletions or duplications, and translocations. You cannot detect mutations or genetic mutations because that is not the purpose of karyotyping. It is also important to know the band resolution. So karyotyping resolution depends on how many chromosomal bands are visible under the microscope. So when you have a lower resolution, you have 350 bands, which is visible on this part of the screen, and this is how it looks. The same chromosome, when it is taken to a higher banding of 550, this is how it looks, and 850 is much more detailed. So a high resolution view of 550 to 800 bands identifies subtle changes like deletions, duplications, translocations, and inversions, which you may not be able to see in a standard view. So if you're suspecting something, and then I think the cost difference between a high resolution and a normal karyotyping is not too much in these days. So it might be it might make sense to order a high resolution view of the karyotyping. So coming to the impact of chromosomal errors in terms of gain of function and loss of function, which typically are more towards, gain of function and loss of function, we usually see in terms of genes, but chromosomes eventually host the gene. So it is important to understand this. So a normal homologous chromosome, you have two pairs, one from your two copies. So you have one from your father, one from your mother, and you have the same gene at the same location of the chromosome. So if both the genes, if both the copies of the genes are producing the polypeptide chain eventually, then you have a normal production of protein, right? But if you have one extra copy of a chromosome, what happens is you have an increased production of a protein and you have a gain of function because these genes are all supposed to be functional. You have 3 copies of one particular chromosome. So you will end up having 3 copies, 3 units of protein which is being coded. Then the missing copy, then if you have one chromosome less, then you end up having a missing copy of a protein. So you have a decreased production of protein. So that is called a loss of function. So keeping this in mind, we will move forward with with chromosomal errors.
1 minute, Doel, can I make an announcement?
Yes, ma'am.
Doel, we are not having the third speaker. She has just had one emergency in the family.
Okay.
So your talk time is increased by 15 minutes.
Okay.
So you can conveniently finish your lecture. And the second session, Jaya Chawla ma'am, I also request her to prolong her lecture, like up to 45 minutes, 1 hour, it's okay. Thank you, ma'am.
All right. Okay. So chromosomal errors. So we will go to first, we all know we have numerical errors, difference between ploidy and SOMI, and structural errors. So if you have to classify chromosomal errors, you will have a numerical error or an error in a structure. So basically, when you suspect that there is something wrong with the patient, you are not going to directly sequence the whole genome and figure out what mutation that person has. What is the best way? First, you will want to count and then look at the structure with the available tests that you have. So numerical error, you have 2 types. One is when you have more than 2 sets of chromosome. And the second is when there is a mismatch on the number of chromosomes. So when you have, so taking the same concept ahead, when you have polyploidy, you have greater than 2 sets of chromosomes, right? So where N, we know N is haploid. Haploid is 23 is haploid for human beings. 2N is diploid, 3N is triploid. So when you have triploid, you have, instead of having a pair of chromosome 1, a pair of chromosome 2, you end up having 3 chromosome 1, 3 chromosome 2, and so on. So your total number of chromosome instead of 46 becomes 69. So 15% of spontaneous abortions is because of the triploidy, and it is the third most frequent chromosomal error. And tetraploidy, where you have 4 sets of chromosome, making the total number to 92. So approximately 2% is much less frequently a presentation as compared to a triploidy. Moving to aneuploidy. Aneuploidy is when you have one extra or one, not one, I mean when you have extra or missing chromosomes. So instead of the entire set, you just end up having either one extra or 2 extra or 1 or 2 less. So 2N meaning a diploid presentation, you're supposed to be a 46 when you count the total number of chromosomes, but instead of that, you are 2N plus minus 1, then you end up being a monosomy or trisomy. So a monosomy is when you have one missing chromosome, so you have total 45 chromosomes. So monosomy X is the most frequent first trimester loss. Trisomy is when you have one extra. So here, instead of 46, you have total 47 chromosomes. So approximately 20% of the first trimester spontaneous miscarriages is because of trisomy. Moving to structural rearrangements. So structural rearrangement are of 2 types. One is unbalanced and then there is called a balanced. So what happens in an unbalanced is the total amount of genetic information changes. So either a segment is deleted or a segment is added. So your genetic information has changed. That is why we will call it unbalanced. So when there is deletion, there is a loss of information, so loss of function. When there is addition or duplication, there is gain of information or gain of function. In case of a balanced rearrangement, the total amount of genetic information is going to remain same. It is just going to be at a different place. So when we talk about unbalanced, we are talking about deletion. Deletion is a segment deleted. When we talk about duplication, you have a segment duplicated. So you have double amount of genetic information here. When you have insertion, you have one segment of a chromosome inserted at some other place at another chromosome. And when you have a ring, you end up losing the telomeres and it forms a ring. So it is also considered an unbalanced because you end up losing some gene with the telomeres. Now coming to balanced rearrangement. So in balanced rearrangement, you have inversion. What happens in inversion is the genetic material is same. It's just that it is inserted upside down. So it again is a problem because there are, reading becomes difficult when you are reading an inverted gene. So it is called a balanced because the information, net information stays same. Translocation is when a segment is translocated. This is where you have 2 kinds of translocation, a reciprocal and a Robertsonian. Reciprocal translocation is where one segment from one chromosome is translocated to another chromosome, and wherever it is translocated, that particular area is being, attached to the previous. So basically this segment has gone to the next second chromosome, and whatever was supposed to be here, which is this part, is now attached to the other chromosome. So there is reciprocal exchange of information. In Robertsonian's translocation, the entire chromosome attaches to other. So these are 2 acrocentric chromosomes. They have attached with one another, and this is what a Robertsonian's translocation is. So moving further, So before this, I think initially I had it planned because let us move to genes and then we will this. All right. So I hope bit about chromosomal errors is clear. Now moving to the actual genes and genetic mutation. Like I mentioned, this we already know, these are nucleosomes. Nucleosomes are screen pointers. Okay. Nucleosomes have DNA bound to it, and these nucleosomes will hold tightly a DNA, and the DNA has genes on it. What is important to understand here is not everything written in gene is a genetic code which will be read, transcribed, translated, and will eventually form a folded protein. There are many segments on the DNA which do not have coding sequences. So the gene can be divided into exons and interons. An exon is the coding sequence and interon is a non-coding sequence. Actually, you have more non-coding sequences than coding sequences. How I try to remember exon and intron is coding sequences with an X, extrovert is coding it. This is how I try to remember that. Intron is introvert. So those are non-coding sequences. So genes will have many non-coding sequences. Now, what is a locus and what is an allele? Gene's position on a chromosome is its locus. So a particular gene, which for example is coding for, say, your eye color, that will have a particular position on your chromosome always. So that is called its locus. And different versions of the genes are called alleles. So like we saw that, there are multiple codons which end up giving the same amino acids. So which means that if we want one particular product of a gene, there may be more than one way of reaching there using different combinations of codon. So that is what different versions of a gene mean. So different versions of a gene are called alleles. So allele are like we see, I think this Punnett squares and the diagrams we have done in our 12th standard where a dominant trait is a capital, a recessive is a small a. So this is what we have done. A heterozygous is when you have, a capital and a small mixed, and you have homozygous when you have the same versions together, like capital A's and small a's. Now, what is gene mutation? Changes in the structure of a gene, which is the sequence and the number of the nucleotide or a codon, without changing its position, is called a gene mutation. So the effect of a gene mutation could be either loss of a function, like we saw what is a loss of function. It could be a neutral mutation, or it could be a gain of a function. We will come to it in a while. So changes in the gene or sequence and number of nucleotide are called gene mutations. So when the mutation occurs in the protein encoding sequence, it impacts the protein synthesis. When it occurs in the transcription regulating sequences, it impacts the genes interaction and genes regulation. So it's gene interaction is when one gene product is going to ask some other part of the gene to, start production of transcription. So it's a complicated structure where one, it's like a, how do I explain? It's like a switch where you start one thing, it gives something, and that something is going to code for something else. So that is what gene interaction and regulation is. Now, gene mutations are alterations that occur in DNA sequence from a single nucleotide to a size up to 100 kilobyte. So genetic mutations can be either a point mutation or a repeat expansion. So we have to now understand what is a point mutation and what is a repeat expansion. I think I will have to go back to my other presentation where I have the entire genetic. I was not prepared for giving this, but I will quickly go to that. Just give me a few seconds and I'll be back with the genetic mutation lecture. Mutation. Yes.
So I also advise all my viewers to please jot down a few questions. If you have any queries at the end of the lecture, Dr. Doyle will be taking those queries. So you can always ask your doubts.
Okay. So point mutation is where you have either a substitution of a nucleotide base or you have insertion or deletion of a nucleotide base. And these base substitutions can be of three types. They can be either a silent mutation or a missense mutation or a nonsense mutation. Let us see how. Like we saw that a codon GAA is going to code for a glutamic acid, right? Now, due to a mutation on the gene, if the GAA, the last alphabet A, is converted to a G, we will refer a codon wheel to see what happens. So we have a GAA and a GAG, both coding for the same amino acid. So even if your mutation has changed, knocked off an alphabet A and changed it to a G, you are ending up with the same amino acid. So the net effect is that it is a silent mutation. There has been no change in the amino acid. So it is a code redundancy and that person will have no issues because you end up coding the same gene product. The next one is a nonsense mutation. What happens in a nonsense mutation is if the first letter has changed to, for example, a G has changed to a U, you were supposed to code for a glutamic acid. Let us see on a codon wheel what is a GAA to UAA. So instead of a G, you had a base substitution of a U. So UAA ended up being a stop codon. So instead of assembling a polypeptide chain, one amino acid after the other, the assembly is going to stop. The transcription is going to stop and the synthesis is going to stop prematurely. So this becomes a nonsense mutation. And then there is something called a missense mutation, where the change in the base substitution has led to, let us see what was the change. So instead of GAA, if your last letter changed from an A to a C, so GAA was supposed to be glutamic acid, now you will be coding for aspartic acid. So you are coding altogether a different amino acid. So this is a huge error. So a missense mutation has changed the final protein. So this is going to present as a disease. So examples of point mutation. So for Marfan syndrome is one of the nonsense mutations that we can study. This is an example which is, highly quoted in many genetic textbooks. So tyrosine, instead of coding for a tyrosine, the person ends up coding for a stop signal. That's why it is called a nonsense mutation. So the protein production will stop, before.
Doyle, your slides are not running.
Are they not? I am running the slide. Are they not?
I think we are on the sickle cell anemia slide.
Really?
Are you
Okay.
seeing the slides moving, Purva?
No, ma'am, we are on sickle cell anemia slide.
Oh, yeah. This presentation is not started only, Doyle.
Okay, let me start that again. Let me
Yeah,
start.
I think stop share and start share again.
Yes, I'll do that.
Because our screen is the fixed on sickle cell anemia missense mutation.
Okay. Can you see the screen now? Shall I repeat what I just, because otherwise it won't make sense.
Yeah.
Can you see the screen now?
Yeah, it is genetic mutations, point mutation, and base substitution.
So the base substitution is where a nucleotide base pair is substituted by some other letter. There are three kinds: silent, missense, and nonsense. What happens in a silent mutation is the change in the base where here the letter alphabet A is changing to G. So instead of coding for glutamic acid, it is okay. So instead of GAA, if the mutation ended up changing the last letter to G, we see that glutamic acid have two ways of being coded, GAA and GAG both. So even if there was a mutation, you ended up coding the same amino acid. So this is a silent mutation. So there is no net impact. If it is a nonsense mutation, so instead of G, if a mutation ended up adding a U instead of a G, so in net change, the GAA to UAA will end up coding for a stop signal. Stop signal means the transcription stops. There is no further codons which are being transcribed. And hence, the net impact is you don't end up producing the glutamic acid and it prematurely stops. So it is called a nonsense mutation. And missense mutation is where the change in the base is going to end up coding for altogether a different amino acid. So the change of GAA to GAC. So GAA was glutamic acid, but GAC is aspartic acid. So you end up coding for altogether a different amino acid, and this is going to manifest as a disease for a missense mutation. So examples of point mutation are Marfan syndrome, which is a nonsense mutation. Here, tyrosine, instead of coding for tyrosine, you end up coding for a stop signal. So tyrosine of the fibrillin 1, FBN1 is the name of the gene, is transferred to a stop codon. And the synthesized fibrillin protein is shorter than normal. So normally, whatever the size is, because of the stop signal, there's a premature stop. You don't end up coding the entire fibrillin protein. And this represents manifests as a disease in the Marfan syndrome. In sickle cell anemia, which is a missense mutation. So missense, you end up coding for altogether a different amino acid. So just one change in an alphabet. So instead of a CTC, there is a CAC. So instead of coding for a glutamic acid, you code for a valine. And this change manifests in a sickle-shaped red blood cell. And I am sure in the next presentation, we are going to hear more about it. So now going to what are indel. So what we saw was base substitution. 3 kinds of problems of genetic mutation occurring because of change in one base nucleotide base pair. Indel mutations are insertion or deletions. So indel mutations are of 2 kinds, frame shift or the ones which are in multiples of 3 nucleotide base pair. So frame shift mutation is where your reading frame changes. So instead of reading, TTA, TTC, you change you shift the frame to the next one because of a mutation. I have to put the pointer again. So because of some mutation, your reading frame completely changes, right? So this causes entirely different amino acids be coded. And okay, a frame shift mutation in a gene refers to an insertion or deletion of nucleotide base pair in number that are not multiples of 3. So for example, if you have just insertion of 1 base pair, right? Then your reading frame has changed. But if you have insertion of 3 base pairs, your reading frame remains intact, but you end up coding 1 extra bit of amino acid in between. So original reading frame is CCU, GAC, UAC, and CUA. But due to a mutation, which is deletion of 1 nucleotide of U, which is at this position, you end up reading completely different reading frame and you end up coding for a new and entirely different polypeptide chain. So this is called an indel mutation due to a changed reading frame. So beta thalassemia is actually a frame shift mutation. More than 250 mutations have been identified on the beta globin gene on chromosome 11. So beta thalassemia is caused by reduced or absent synthesis of beta globin chains of the hemoglobin tetramer. So this is hemoglobin, this is the tetramer. So there are two alpha globin chains, two beta globin chains. What happens is this is present on the HBB gene, chromosome 11, which is supposed to be coding for the beta globin, right? Due to a frame shift mutation on the beta globin, which is, for example, missing here, this gene is missing beta globin gene, you end up creating, okay, there is a point mutation due to which there is a change in the reading frame and it will present as a beta thalassemia. Gene mutations are alteration. Okay, now we are coming to repeat expansions. So trinucleotide repeat expansion or TNRs are a normal gene with CAG repeats. So when you have, this is a normal gene with one CAG here, two CAG, three CAG. So these are CAG repeat segments, but in a trinucleotide repeat of CAG, you end up having too many CAGs inserted, and this ends up causing a hairpin-like structure. I think, okay, trinucleotide repeat expansions could be due to coding region repeat, which is Huntington's disease, which presents as a CAG repeat, and Kennedy disease, which presents as a CAG repeat again. Non-coding repeat region repeats are Fragile X, which is a CCG and CGG repeat, and myotonic dystrophy, which is a CTG repeat. So genetic disorder classification. So either you have a monogenic or a single gene which is mutated or disorder. That is why you have the disorder. So they are called monogenic. Polygenic are when you have multiple genes involved. Mendelian disorders are monogenic or single gene disorders. There are about 7,000 known disorders which affect over 400 million people worldwide. So these are only known. There are many which we are not even aware of, where research is still going on. So these are called Mendelian disorders or monogenic diseases. And these are the ones we actually test for when we do our PGTs. There are, though, a couple of polygenic diseases which these days new panels have come up for them, but I'm not exactly sure as to about their accuracy. I think geneticists and people who work on these labs will be able to tell more about that. So this brings me to the end of my presentation. So these are my references. And I think that the chromosomal bit was okay, but when you actually come to the genes, it's very difficult to understand the genetic mutations until you keep, in a short frame possible time. You have to read it again and again to understand that. But once you understand that, it becomes very easy to understand what kind of test and how to go about it. So, and epigenetics is beyond this lecture. It is a lecture in itself.
So, chairperson's comments, and Dr. Randhir is also there.
Yeah. Didi,
Shaban,
I'm audible.
yes,
I'm
yes,
just
Shaban.
driving the car, so I'm not opening my video. Ms. Joel, it's an excellent presentation, quite elaborative, and I think you made it very simpler. So I really appreciate this talk. Thank you so much.
Dr. Randhir, are you there?
Yeah, Dr. Doyle, you spoke very well, and I think you are the best person to give this talk. And I have heard you many months before also when we started, and you improve on going on adding on that. So I think next in genetic webinar, you will add a few more things. And this will be very useful for our IVF ART, because epigenetics are very important. A lot of in the future, though the 35 years, 1978, we have got more than 35 years baby. So I think there is no doubt, but people still say, is there any genetic difference and all? So very nice, and hope we will be taking you again your knowledge in the webinar. Thank you.
Doyle ma'am.
Yeah, Jagriti is there. Hello, Jagriti.
Hello ma'am.
I hope you enjoyed the lecture.
Yes, thoroughly enjoyed ma'am. It's very informative, enlightening talk given by Doyle ma'am. I thoroughly enjoyed the talk. It's beautiful ma'am.
And Jagriti, you're always there in so many of my programs. So thank you for always supporting. And
For
whenever
giving me opportunity.
whenever we think of Sagar, we think of Jagriti. So, Doyle, it was another great show. Just little suggestion. I do appreciate all your slides and all the high-tech, the videos which you showed us, the uncoiling of the helix and everything. If with every disorder, like the point mutation, the missense mutation, the CAG sequence, you could put in, just flash the name of the disease associated. That will bring a lot of, clarity to the, thought process of clinicians, because we'll always remember, oh, point mutation is this disorder. Single gene disorder is this one. Balanced translocation is this one. So in your slide, just a, little pop-up would be really, it'll make things even, it'll take them to another level. So thank you so much, Doyle. And we've given you more homework to prepare the sequelae to this so that we can call you again. Whenever you're ready for your sequelae, we'll create a program for you. Thank you so much. Over to Dr. Shweta for session 2.
Yes, so session 2 will be taken up by Dr. Jaya Chawla ma'am. Can I have the CVs in session 2 chairpersons? Okay, the topic is screening of hemoglobinopathies, and session 2 and session 3 will be continued by Dr. Jaya Chawla ma'am. And session 3 will be on 11 to 14 week screenings and aneuploidies. And we have chairpersons Dr. Monica Singh ma'am. Madam is President ISOPAP Society Bhopal chapter. Madam is National Executive Member of ISAR. And
Shweta, you can skip this.
We welcome you, ma'am. And we have Nidhi Jain, ma'am. Madam is Secretary, Sagar Society, Obs and Gynae. Ma'am is a avid practitioner. She has been practicing for more than a decade, and she also spent a lot of time in patient counseling and spreading education and awareness to the patients. We welcome you, ma'am. Next, we have Dr. Kavita Bhati, ma'am. Madam is Professor in CMC Hospital, Ludhiana, Punjab. Madam is Vice President, Ludhiana Obs and Gynae Society, President, Ludhiana Menopause Society, and President, Society of Vaginal Surgeons of India. She is also Secretary, Punjab Chapter, North India Gynae Forum, and past member of Medical Education Committee and Medical Practical Obstetrics Committee of FOXI. And Madam is editorial board and peer reviewer and co-editor in many journals. And she has been awarded as Best Clinical Teacher Award in 2017 Wonder Foxian Award. We welcome you, ma'am. Next, we have Dr. Professor Aruna Verma, madam. Madam is Professor at Department of OBGY LLRM Medical College, Meerut. Madam is has a vast interest in endoscopy, and she has taken training in from AIMS New Delhi. She has also done training from Fox Recognized Advanced Gynae Endoscopic Training Centers, and she has done fellowship from WLH in 2023. She has been awarded as the Best Research Paper Award in UPCOG 2018 and awarded a gold medal for Best Paper Publication in Maternal Health of Rural Women category in UPCOG 2019. She is the peer reviewer, co-editor, co-investigator, and of ICMR projects. We welcome you, madam. And lastly, we have Dr. Professor Preeti Jindal, madam. Madam is Director, Touch Clinic, Mohali, and also Director, Robotic Gynecology Park Hospital, North India. She was the past president, Mohali OBGY Society, General Secretary, INSAR, Senior Consultant in Gynecologist in Fortis Hospital as well. And Madam has been awarded as for being one out of 50 most influential Indian women. And she has written many books. And she is also the main editor of Aesthetic and Regenerative Gynecology, and also the Young Forever Menopause book she has written. So we welcome you, madam. Over to and next we have Dr. Jyoti Chauhan, madam. Madam is President Sagar Society, and she has also guided her team to bag 4 coveted national awards. And she's also the fellow of International College of Maternal and Child Health. And she has presented multiple papers and national international conferences. And she has also invited faculties in national and state conferences. We welcome you, ma'am. Over to chairpersons. Dr. Jaya Chawla, ma'am, CV kindly show.
Who is introducing Jaya, madam? Dr. Aruna is there?
Yes, ma'am. Aruna madam is there.
Aruna, madam, are you there?
Ma'am, you can start.
Okay, so I will take the pleasure of introducing Dr. Jaya Chawla. Welcome to Synergy 15, madam. And we are very, very delighted to have you amongst us. And thank you for doing the third session extempore as well for us and covering up for Dr. Deepika. So I would like to introduce Dr. Jaya Chawla, who is very active in the Society of Fetal Medicine in Delhi chapter, where she is joint secretary. She is also the executive member of Genetics and Fetal Medicine Committee of FOXI, executive member of AOGD, Fetal and Genetics Committee, and Safe Motherhood Committee, AOGD 24 to 26. To her credit go the accolades of co-editing the FOXI focus on RH isoimmunization in 24, editor of OSCE in Fetal Medicine and Genetics for FOXI GFMC 25, and editor of OBGY clinics on renal disorders in pregnancy for AOGD in 25. She's also contributed to FOXI GCPR on stillbirth in 23. She is the dissertation supervisor for PG DNB students and super specialty courses, examiner for postgraduate and NBE courses. And as for the research, her profile goes, she's member of IRP, investigator of ICMR projects with several publications in national, international, and index peer-reviewed journals and chapters in books. Her fields of interest are fetal cardiac arrhythmias, prenatal screening, and fetal growth restriction. Hello, Dr. Jaya Chawla, and we are really honored to have you amongst us.
Thank you, Ma'am Monica. It's always a comeback to Bhopal FOXI, and I'm really, really delighted to be a part of this all over again. I also thank ISOPOP Bhopal chapter and the Sagar OBGY Society for having me here. And I seek permission to share my screen, please.
Sure, Dr. Jaya.
So is this visible, please?
Yes, your screen is visible.
So I'll be starting off with screening of hemoglobinopathies. That is the first agenda for today. As we all know, this is a brief take through the embryology period and the composition of various hemoglobins in the human body. So hemoglobin F, which constitutes a major kind of hemoglobin from the fetal weeks of 16 weeks to say 28 weeks, after which it starts getting reduced. Adult hemoglobin, which takes over at somewhere around 28 to 30 weeks. And from birth onwards, it constitutes an increasing level of adult hemoglobin. And finally, we have hemoglobin.
Can I have 1 minute? One
sure,
minute, Dr. Jaya. Support team, there is a band which is coming over the slides. There is a gray band which is coming on top of the slides.
Madam, that must be support team. Can we manage this?
Yeah, there is something coming
It is
on.
your laptop's problem.
No.
Is it?
Dr. Jaya,
Yes, ma'am. There has been.
Is there a gray band on top of your screen? So it is hiding the head.
No, it is not. Like it's a usual Zoom meeting that we have, the audio video kind of thing that we have, the bar that we show on the top.
Are you sure? Are you
Yeah,
sure?
yeah, absolutely. This is the usual thing that
Yeah. Support team, are you sure it's nothing from our end?
Yes, ma'am. It's nothing from our end.
So I think we can
just reduce the minimize the there is a minimize button. If you can just do that, maybe it will go on the side instead of having there is a small hyphen like
There's a view thing. Yeah, let me check that part out. No, now this is gone.
Yes, now
Is it better now?
It's better now. It's become very small now.
Yeah. So perfect. That was the bar, I think. The Zoom bar that we have on top. That was that.
So good. We are good now. Thank you.
Perfect. Perfect. So now we talk of the composition of various hemoglobins to be able to understand why thalassemia is going to be a matter of concern, and then what will be the pathologies in terms of alpha and beta thalassemia. So when we talk of fetal hemoglobin, the composition is alpha 2 and gamma 2. I'll be coming to the compositions in detail in the subsequent slides, but here it suffices to understand that hemoglobin A, the adult hemoglobin, is alpha 2 and beta 2, and A2 constitutes a small percentage, alpha 2 and gamma 2. So what you realize is that when we do have a problem with the beta chain, the adult hemoglobin gets reduced, and therefore beta chain abnormalities will cause significant maternal hemoglobinopathies because a major chunk of the adult hemoglobin will be affected. But when we see that alpha chains are a component of all of these three types of hemoglobins, we tend to understand that right from the fetal period itself, there will be a concern if the alpha chain is affected. And that is why alpha thalassemia presents in fetal life and beta thalassemia will have problems with the maternal aspect of the maternal fetal medicine spectrum. Now, the genotype versus the phenotype. We need to understand that adult hemoglobin that we say is alpha 2 and beta 2, it constitutes chains from two different components, alpha and beta. But genetically speaking, we have 4 alpha chains coding for these 2 beta, 4 alpha genes which constitute the composition for the 2 alpha chains, and there are 2 genes decoding for the beta chains. So effectively, a mother will have 4 alpha genes which will give rise to 2 alpha chains, 2 beta genes which will give rise to 1 beta chain. The father will have a similar contribution, and effectively 2 alpha genes and 1 beta chain comes from each parent. This is the structure of the hemoglobin molecule. We have alpha chains and we have beta chains, and there is interspersed heme units with iron atoms. So now when we look at the hemoglobinopathies map of the world, we realize the significance of this topic being discussed today evening. The entire Southeast Asia belt is a hemoglobinopathies endowed zone. And when we zoom in on India, we have to understand that you see this green aspect covers a major part of north, west, south, and also east. So this is beta thalassemia. Complementary to it is also the sickle cell disease, which you see interspersed as yellow in almost the entire country, barring the northeast. And northeast is marked by this another variant of green, the hemoglobin E disease. It is important to understand these ethnicities because they constitute the first line of identification of these hemoglobinopathies when the patient walks into the gynae OPD. Coming to what all we're going to discuss today. So a beta thalassemia, we'll be discussing major, minor, and also hemoglobin E varieties. Alpha thalassemia, a bit about that. And we shall also be discussing sickle cell syndromes, including sickle cell trait, disease, and some compound heterozygous states such as S beta thal or S D Punjab. So now when we come to types of beta thalassemia, there are 2 primary types that we need to understand. Again, we need to revise that the lower panel here consists of the 2 alpha chains and that panel on the top consists of 2 beta chains. So now when 1 beta chain goes missing from the top, we still have 1, remember that. This gives rise to what we call as non-transfusion dependent thalassemia or beta thalassemia minor. When both of these 2 chains, the beta chains, are gone, what gives rise to the disease is a beta thalassemia major or a transfusion-dependent thalassemia. So the sequence in the clinical perspective would be, when a woman comes to you, you check the ethnicity, you look at a complete blood count, and if you suspect basis of the complete blood count that she should be a case of a hemoglobinopathy, you offer some special tests, which could be screening tests or confirmatory tests. Once you have arrived at a confirmation of a certain hemoglobinopathy, you will need to understand the molecular basis of that particular disease by offering her DNA testing. And until we have done that, offering a prenatal diagnosis to this lady will not be possible. So coming to the blood picture, we talked of ethnicity. The blood picture, we all understand that microcytic hypochromic anemia is a common factor underlying iron deficiency anemia and beta thalassemia trait. So in this particular table, we realize that beta thalassemia trait, which we like to focus on, but also hemoglobin Laporte trait and delta beta thalassemia trait. All of these are marked by what we call microcytic hypochromic anemia. And how do we identify it? We look at these 2 indices on the complete blood count picture, the mean corpuscular volume and the mean corpuscular hemoglobin count. So when we say MCV, The normal ranges are between 80 to 100 femtoliters. So anywhere when you see an MCV less than 80 and an MCH less than 27, please remember these values, because as I take you through different questions and different quizzes, these will come in handy to understand that the differential of microcytic hypochromic anemia is going to be beta thalassemia minor. Please also concentrate on the lower panel now to see that a persistence of fetal hemoglobin, sickle cell trait, D trait and E trait will be marked by normal MCV and MCH values. So microcytic hypochromic anemia is going to be differentiated between iron deficiency and beta thalassemia trait. Now, until we go to HPLC and decide whether a woman is iron deficiency anemia or beta thalassemia, we have a CBC report in front of us most of the times in pregnancy. And this has indices called the MCV and also the RBC count. So if we take a ratio of the MCV to the RBC count, which is called as the Menzel's index, we are able to, in certain cases, identify that if the value is more than 13, we are dealing with iron deficiency anemia. And if the value is less than 13, we are probably looking at a case of thalassemia minor. The reason being this, that in iron deficiency anemia, the RBC count gets reduced very significantly, but the mean corpuscular volume is not affected as much, even though it is reduced. But in cases of thalassemia, the volume of individual RBC is going to be reduced to an extremely great extent, and the number of RBCs will not be affected as much. And therefore, the ratio would stand at less than 13. Coming to the peripheral smear, we have microcytic hypochromic blood picture along with nucleated RBCs in beta thalassemia major, as you can see. And now we come to a basic screening test for thalassemia, that is the Nestroff test, also known as the naked eye single tube red cell osmotic fragility test. The basis of this test, we use buffered saline here, 0.36%. We see that there is a black line on a paper placed behind the test tube. The premise of this test is that whenever RBCs from a person with thalassemia trait are introduced to buffered saline, they resist being lysed by the saline, whereas RBCs from a normal individual are going to lyse and therefore result in a clear solution. You'll be able to see through the clear solution, and therefore this is a negative test. But RBCs in this particular case will resist lysis by the buffered saline because these are thalassemia trait RBC. And therefore, when you're not able to read the line or the text behind the test tube, it is supposed to be a positive test, and this person should be investigated further for thalassemia minor. It has an excellent sensitivity test for low resource settings. Specificity is okay at 85%, but remember that the positive predictive value of this test is only 66%, and therefore a positive Nestroff is not equal to thalassemia minor. And one always needs to confirm with a confirmatory test. The confirmatory test is going to be what we call high pressure liquid chromatography, high performance liquid chromatography, or HPLC. So here in front of you on the screen is an HPLC report, a normal blood sample. This has been placed here so that we can learn to interpret what to do with the HPLC report. Here we have something called area percentage and something called the retention time. It is important to understand these informations to be able to decipher the HPLC reports better. The intention of putting the retention time here is basically to identify which peak represents which kind of hemoglobin. We have already seen in the initial part of this talk that we have hemoglobin F or the fetal hemoglobin, A2 or the hemoglobin A2, or we have A0, that is the adult hemoglobin. So where are we going to find these peaks? It will be determined by the retention time. So for example, when I say F has a retention time of 1.15 minutes, I go to this bar here, and at 1.15, I find a small peak which represents hemoglobin F. When I say A0 is at 2.36, I mean to say that on this bar, I have to go to 2.36, and the peak I'm seeing here represents adult hemoglobin. So on for A2, that is 3.68, a small peak at 3.68. So this is the retention time telling us the peak which we need to know. But in order to decipher the disease that this particular sample is showing, we have to look at the area, because area represents the concentration of that hemoglobin in the person's body. And therefore, when I say F is 0.4%, A2 is 2.8%, but A0, that is adult hemoglobin, is 88%. This is exactly what we need to decipher the information regarding the hemoglobinopathy status of the patient. Now, basics again. We need to have a relative idea of the proportion of various hemoglobins in the HPLC report to be able to decipher it better. So for all practical purposes, a normal adult will have majority of hemoglobin being constituted by HbA or the adult hemoglobin, and that will be to the tune of 94 to 96%. HbA2 will be between 2 to 3.7%. Usually we take a cutoff of 3.5% to say HbA2 to be abnormal. Anything above 3.5 should be viewed with concern. However, some textbooks mention that 3.5 to 4 is equivocal, and something above 4% shall be taken as abnormal. Absolutely. So still, for clinical practice, we use a cutoff of 3.5% for HbA2 to be normal. Anything below it is fine, above it is not fine. Should be viewed with concern and investigated further. When we come to thalassemia minor, we see that not only A2 rises, but also hemoglobin F rises to a certain extent, obviously at the cost of HbA, because beta chains are deficient, and therefore thalassemia minor will have fall in adult hemoglobin, a rise in fetal hemoglobin, and HbA2. Remember, please, that fetal hemoglobin will rise only to the extent of still being less than 5% in cases of thalassemia minor. For thalassemia major, again, it is very important to note that rise in HbA2 was giving us an idea about thal minor. But an astronomically high A2 is not going to mean thalassemia major. Thalassemia major is constituted by an astronomically high fetal hemoglobin. And it is fetal hemoglobin which is resistant to lysis. So it is important to remember these values as we go further, deciphering some more HPLC reports. So now here you have a North Indian woman whose CBC is marked by microcytic hypochromic anemia, and the peripheral smear shows anisocoagulocytosis. On the top right corner of the screen, you see that the MCV is less than 80 and MCH is less than 27, which means we are dealing with microcytic hypochromic blood picture. The HPLC report says adult hemoglobin is 84.8%, hemoglobin A2 is 6.4%, and fetal hemoglobin is 1%. So here we are trying to imply that a figure more than 3.5 is present for A2, and therefore this should be thalassemia minor. On the adjacent side, the report suggests adult hemoglobin to be very low at only 5.7%. Hemoglobin A2 is 2.5%. Remember, thalassemia major, hemoglobin A2 was not going to rise very astronomically, and fetal hemoglobin is 90%. So we imply that the picture on the right is suggestive of thalassemia major, and that on the left is suggestive of thalassemia minor. Now, it is important to understand that when we have rules in science, we also have exceptions in science. And therefore, increased HbA2 is not just significantly associated with beta thalassemia, but also hemoglobin E, hemoglobin Lepore and Iran in rarer cases, sickle cell disease, megaloblastic anemia, and hyperthyroidism. It is extremely important to bear these differentials in mind for the purpose of clinical practice. Whenever there is iron deficiency anemia, there will be a falsely low HbA2. Again, this can have clinical implications. Also, alpha thalassemia and G6PD deficiency are associated with a falsely low HbA2 value. And therefore, all these differentials should be borne in mind when we are dealing with an HPLC report. Coming to the inheritance, Thalassemia, as we all know, has an autosomal recessive inheritance. So if one partner is a carrier and the other partner is absolutely normal, then we see that the alleles that we get from this partner, these offsprings are going to be carriers. So 50% of individuals here will be carriers as offsprings, and the other 50% will not even be carriers. Remember that thalassemia minor is something that is totally compatible with a great life, and we do not need to offer prenatal diagnosis for an instance where any one of the partners is thalassemia minor. And therefore, if the woman coming to you is thalassemia minor, you must get the partner tested for HPLC. And if the partner turns out to be not a carrier of thalassemia, neither a thalassemia major case himself, then we do not need to offer prenatal testing for the offspring. There is absolutely no reason to cause an alarm in such cases, and they can go as normal pregnancy. However, if the partner is also a carrier and so is the woman in question, then there is a 25% chance that one baby will be 25% chance of an offspring being affected with thalassemia major, and that becomes a life-threatening disorder, and therefore one needs to offer prenatal testing to identify if the offspring being carried by the woman in the womb is a thalassemia major, in which case pregnancy termination would be indicated. Remember, 50% of these will be carriers and they can absolutely continue the pregnancy. There are sometimes some instances where the labs report even these carriers as pathogenic. And therefore, not just looking at the report of a pathogenic or normal variant, you have to look at the mutations to be able to see if the baby in question is going to be affected by thalassemia major or not. And we shall be coming to some such reports in the subsequent slides. Remember, we talked of a DNA testing for confirmation of the molecular basis of disease. So we must understand there are more than 100 mutations associated with thalassemia. So most commonly in the Southeast Asian population, 5 common genetic mutations have been identified, and most laboratories are able to identify them with ease in the parents. And until and unless you have identified the mutation in the parents, you cannot offer a chorionic villus sampling or a placental biopsy to the lady to identify the status of thalassemia in the offspring, because you cannot perform an HPLC on the CVS sample. The molecular basis of disease must be known. We come to see in clinical practice that women are referred at 12 weeks gestation saying, मेरा पिछला बच्चा thalassemia major है और मुझे गाइड किया गया था कि 12 weeks पे जाकर के मैं CVS करवा दूं. Please try to understand that for CVS to be offered, one has to understand the mutations of the parents, and pregnancy is a time-bound condition. Therefore, one must utilize the inter-pregnancy interval to identify the mutations in the parents, and then only prenatal testing in a subsequent pregnancy can be offered. So, of course, CVS can be offered once we have the mutations in place. And now, quiz time. So, we have this father who is a thal minor, and the mutation is given here. We have a mother who is a thal minor, and the mutation is given here. Their first daughter has inherited this combination of mutations, one each from both parents. Their second daughter has only one mutation, as we see, inherited from the mother. So the question here is, what is the diagnosis for daughter 1 and daughter 2? As you can see, daughter 1 has inherited one allele, diseased allele from both the parents, and therefore daughter 1 is a thalassemia major, whereas daughter 2 has inherited only one pathogenic allele, and therefore she becomes a carrier for the disorder, but she is not a thalassemia major, and therefore she can lead a perfectly normal life like her parents. Now, the index pregnancy for which the prenatal had been offered has again a combination of 2 pathogenic alleles, and therefore is an affected fetus which needs to be terminated. Coming to sickle cell screen, Again, sickle cell is also subject to a solubility test like we did for thalassemia. And here the solution is different. It is not buffered saline, but it is sodium hydrosulfite. The premise being the same. Hemoglobin S from the lyse erythrocyte precipitates, causing the solution to become turbid. So if the solution becomes turbid and you're not able to see the lines at the back on the paper, then it is a positive test. And if you're able to see through the solution, then it is a negative test. Of course, there are false negatives. When the concentration of hemoglobin per se is very, very low, as in severe anemia, or the proportion of sickled cells in the entire gamete is less, to the tune of less than 10% or so, and there are high levels of concomitant fetal hemoglobin, then you're going to face a condition where the solubility test for sickle cell will be falsely negative. And this needs to be borne in mind. In sickle cell, you have an extremely characteristic peripheral smear picture. It's going to be microcytic hypochromic anemia with nucleated RBCs, with polychromasia, but you will see typically these sickle cells, as you see on the slide. Hard to miss nowadays. Earlier, when sickle cell used to be not so commonly discoverable in India, these were not understood properly. But now we have a clear understanding of what the peripheral picture for sickle cell looks like. Here, the adult hemoglobin, I'm talking of a sickle major, homozygous, what I mean. Adult hemoglobin is going to be very low and 0 to 30%. A2 is going to be 3.5% or low. Sickle hemoglobin is going to constitute more than 50%, and hemoglobin F is going to be a little less than 50%. So this information is going to come in handy when we look at the HPLC reports for sickle cell disease. Coming to the inheritance, it is the same as thalassemia minor, and therefore when both partners are carriers of sickle cell, we expect 25% of the progeny every single pregnancy. We expect 25% of individual offsprings to be affected by sickle major or homozygous condition. And remember, ऐसा नहीं है that if a woman is going to produce 4 children and one has already been affected, the rest of the 3 are going to go scot-free. No. Every single pregnancy, she stands at 25% risk of having an offspring, which is going to be affected by the major form of the disease. And therefore, every single pregnancy, prenatal testing will have to be offered. Now, coming to this HPLC report, we have learned to look at the values of fetal hemoglobin and A2, 0.8%, 4.4%. So if we just look at a tubular value of A2, saying more than 3.5% is equal to thalassemia minor, then we are going to miss something here on the top. That is the S window, 4.51 retention time. You see this peak here, which is not commonly found in all HPLC reports. And this is 34.4%. So this is clearly a case of sickle disorder and not thalassemia. Here you need to understand that if it is between 20 to 40%, the sickle component, then it is a heterozygous or a carrier state kind of thing. And if it is to the tune of 70 to 90% in the entire contribution, then you are going to look at homozygous disease. So now look at this HPLC report, which says adult hemoglobin 44.7%, A2 is 4.2%, and HbF is 0.4%. Again, only looking at this much should at least, raise an alarm that this does not total to 100%. So what is the component that is missing that we are not able to see? We are not able to see that the sickle window is also showing a contribution of 45.6%. So obviously this is a case of sickle minor. So if Mrs. VP, the lady in question, has this HPLC report and the partner is also sickle 35.3%, then we are basically looking at 2 partners who are carriers of a sickle cell trait. Subsequent slide, we are going to show you the HPLC report of the offspring. Here you see that A2 is again 4.4%, but sickle window is 91.2%. Which means the offspring is affected by homozygous sickle cell disease. The question is, in what percentage of cases is this offspring likely to have this kind of an outcome? Is it 25% or it is 50%? We know that it is a autosomal recessive inheritance, and therefore 25% of the offsprings are going to face this kind of a malady. Coming to what we call compound heterozygous conditions or sickle cell with thalassemia. Here you see that one partner is carrying a sickle cell allele and the other is carrying a beta thal allele. So again, going by the principles of autosomal recessive inheritance, one offspring out of 4 or 25% of times, we are going to get a condition where the baby is going to inherit sickle from one partner, beta thal from another partner, and become a compound heterozygous individual, sickle thalassemia. The rest of them will either be carriers of one of these disorders or shall be absolutely normal. So now coming to what we call hemoglobin E disease. It is significant because of significant mobility, professional and social, between various geographical locales of the country, results in a lot of northeastern population being a part of our OPD clinics. And therefore, awareness regarding hemoglobin E disease is equally important in clinical practice. Remember that 22 to 80% of population in the northeastern region can carry a hemoglobin E kind of trait. Remember that it is asymptomatic in both homozygous as well as heterozygous condition. So it is not going to cause much of a trouble for the woman in question per se. The problem arises when, because of social mobility, hemoglobin E disease with beta thalassemia come together. So when we have a compound heterozygous condition called as hemoglobin E beta thalassemia, that is symptomatic with moderate to severe anemia, and therefore we need to identify individuals with this combination. HBE per se, both in the minor and the major forms of the inheritance, is not going to raise a havoc in the woman's life, even if she is pregnant. Of course, we have a solubility test where a blue compound is used to identify. Now, inheritance, we all know that all of these are going to be inherited as autosomal recessives, and so I'm not going to the details of these. Here you see that A2 is 38.9%. We said that astronomically high levels of A2 are not associated with thalassemia major. In the beginning of this particular talk, we alluded to this. Now, the question that arises is, why is A2 rising in hemoglobin E trait? The answer is that hemoglobin A2 and hemoglobin E elute in the same window of the HPLC because the retention times are the same. And therefore, in an HPLC report, it is almost impossible to differentiate between whether the contribution is because of A2 or the contribution in that particular peak is because of hemoglobin E. These days we have like rarer kits that can identify, but majority of laboratories will not give you this answer. And therefore, it is important to be aware of the fact that a high level of A2 is going to be associated with hemoglobin E disease and not with thalassemia. Of course, A2 can rise because of thalassemia minor also, and therefore we need to identify whether it is E trait or thalassemia. For E trait, A2 is going to be between 20 to 40%, but F is going to be normal. We have seen in the table earlier that MCV is not going to be low, it is going to be okay. It will not be microcytic hypochromic anemia for hemoglobin E disease, and therefore the patient is asymptomatic. She doesn't have anemia per se. Important to understand that fetal hemoglobin, if it rises to a great extent, then you are not looking at hemoglobin E disease. For E, hemoglobin F is going to be normal. If F is rising, you are dealing with a compound heterozygous E beta thal. So we have seen HBE minor, that A2 is between 22 to 40% and F is normal. If F rises to less than 5%, but A2 goes beyond 50%, very high, then, and also you have a microcytic hypochromic kind of blood picture. MCV is low. With a mild anemia, you are dealing with hemoglobin E homozygous. So now what happens when you have hemoglobin E beta thal? Remember, F high means compound heterozygous. E only, whether minor or major, is not going to be associated with astronomically high values of hemoglobin F. So why is this EE homozygous at F concentration of 4.2%? We said F not more than 5% is associated with hemoglobin E homozygous. So because F is not very high, this is hemoglobin E homozygous and not a compound heterozygous condition. So in order to differentiate between these two conditions, you have to understand that hemoglobin E minor will have no anemia or mild anemia. A2 will be higher in cases of hemoglobin E major, and A2 is going to be extremely high in cases of E beta thal. F is not going to rise at all in hemoglobin E minor, may rise to the tune of less than 5% in hemoglobin E major or homozygous condition. But in E beta thal, F is going to be extremely high. That is the condition you should watch out for, because that particular woman is at a higher risk for anemia in pregnancy and also hemorrhage later. Coming to alpha thalassemia, we have already read that alpha globin genes are 4 in number. So alpha alpha oblique alpha alpha is a normal combination. One gene missing will be a silent carrier. Her life will not be affected. Even 2 can be managed as an alpha thal trait. But if 3 genes out of 4 are missing, then it gives rise to what we call hemoglobin H disease, and these women do have anemia. But if all the 4 alpha genes are missing, that is hydrops fetalis or hemoglobin Barts, this is going to cause a major problem in the fetal life, giving rise to what we call non-immune hydrops, and therefore this needs to be tested. Remember that microcytic hypochromic anemia on CBC, where HPLC is absolutely normal, can be a harbinger of alpha thalassemia. It can be diagnosed only on genetic testing. In cord blood, you can identify this, and intrauterine transfusions can be life-saving, but the baby will be completely transfusion dependent for the rest of his life. Alpha thalassemia, we have realized that this is the composition. Remember that extremely high levels of hemoglobin F are a hallmark of alpha thalassemia, and peripheral smear is going to show you inclusion bodies in places of 3 genes are missing out of 4. So it gives rise to a picture where you have ascites, you have pleural and pericardial effusions, and there's a raised MCA PSV because of severe anemia in the fetus. And all of this you can suspect once you have an Rh positive mother, HPLC most likely normal. That is the time that that means low A2 basically, you should think of alpha thalassemia. This is one condition where both partners should be, the CBC of both partners should be looked at, because both are likely to be microcytic hypochromic for them to give rise to a baby who is going to be hydropic. Genetic testing for thalassemia is mandatory, and once you have the mutations in hand, you can most certainly offer prenatal testing in a subsequent pregnancy. Now coming to clinical pitfalls, I'll be showing you some reports and I'll invite you to take a cue and tell us what they are. Now, this is a lady, 36 years old, a second gravida, has had a previous delivery. She is currently 9 weeks pregnant, Indian origin. She has, as you see here, a normal MCV, normal to low, less than 80. MCH is less than 27. So she has microcytic hypochromic mild anemia, 10.6 grams. In fact, she is in the first trimester, so 11 being the cutoff, this is mild anemia. Her HPLC report says A2 concentration of 5.9%. Adult hemoglobin is 86.4%. So this constitutes a picture of thalassemia minor. But 4 years prior, in her previous pregnancy, her hemoglobin A2 was found to be normal at 2.9% with a hemoglobin of 7.2 grams per deciliter, and she had microcytic hypochromic anemia even then. So she asks, मैं तो thalassemia तब भी होनी चाहिए थी. Why in my previous pregnancy I was not a thal minor, but in this pregnancy I have been diagnosed as a thalassemia minor. Why should that be the case? The answer is iron deficiency anemia tends to lower the levels of HbA2. And therefore, whenever you find a lady with microcytic hypochromic anemia, it is good to give a trial of oral iron for 15 days. And if you do not see a response to therapy, then you can offer HPLC, because lowered hemoglobin values will cause the HbA2 also to be lower, and you might get a falsely normal report of HPLC, even though the woman is actually thiol minor concomitant with iron deficiency anemia. It is good to recall here that megaloblastic anemia is going to give rise to a falsely elevated A2, and therefore correction of anemia by whatever is the disorder identified, whether it is iron deficiency or a B12 or folate deficiency, is important before you diagnose or refute, for that matter, a hemoglobinopathy on an HPLC report. Now here we see that the hemoglobin of the patient is 17.4 grams per deciliter. She's a 22-year-old primary gravida with a family history of abnormal hemoglobin. Her A2 concentration is 44.3%. And of course, she has polycythemia. Now, this does not fit into any of what we have discussed thus far. And this is a variant hemoglobin called as hemoglobin Bethesda. The take-home message here is, whenever you get a blood picture like this, you need to seek expert genetic counseling. And this is not something of the order of a regular obstetric practice. Now it is time for some brainstorming. So we come to a brief quiz kind of a thing. And here you see that we have an adult hemoglobin in this report of 80.5%. And hemoglobin E, there is a peak of hemoglobin E, which is 16.5%. So what are we dealing with? Of course, we are dealing with, they have given you a clear hemoglobin E report. Normally, they say A2. Here, they have given you a separate hemoglobin E report, and therefore, it is easy to identify that this is a case of hemoglobin E minor or hemoglobin E trait. Now, this 33-year-old lady from Mizoram is a primary gravida. Her hemoglobin is less, 10 grams. Her MCV is low, her MCH is also low. So it is a microcytic hypochromic anemia with an anasopoikilocytosis kind of picture based on the increased RDW. Her adult hemoglobin is 59%. Sorry, A2 is 59%. A2. Remember, A2 elutes in the same window as hemoglobin E. And adult hemoglobin is low. Fetal hemoglobin is high. Whenever we have a high A2 associated with a high hemoglobin F, we have learned that it is a condition to be worried about, and we call it the compound heterozygous E beta thal condition. Now here it is pretty clear that it is sickle peak is also there, 33.6%. Because it is between 20 to 40%, we can say that this is a sickle cell trait condition. So the algorithm for testing of hemoglobinopathies is going to be, first you offer a complete blood CBC kind of thing or hemoglobin. If it is low, you offer a solubility test or Nesterov kind of thing. If the RBC indices are also conglomerating with the entire jigsaw and your HPLC gives you an idea about a hemoglobinopathy, then you need to identify the mutation by offering DNA-based tests and counsel the patient for prenatal diagnosis in case the patient and the partner both happen to give you an indication for prenatal sampling. And finally, counseling for termination of pregnancy if the disease in question is confirmed in the fetus. This is a ready reckoner because not every trait marrying with every other trait is going to give rise to a matter of concern. For example, if you look at hemoglobin E disease with sickle, it is a white window, which means not a matter of concern, minimal risk. So instead of scaring the patient that you are also a carrier, your partner is also a carrier, you must go through a reckoner like this to see what exactly is the risk, and it will be best to send such patients for genetic counseling formally, wherein their risks and their methods to allay the risk finally can be discussed in detail, and that professional advice is going to be beneficial not only to the obstetrician but also to the patient. So with this, I come to the end of this particular session. And these are 3 books that we have released for the Foxy Genetics and Fetal Medicine Committee, the ones on the left. and for the AOGD, the renal disorders in pregnancy and endometriosis OBGYN clinics. So we welcome you all to read some of these for more insights. Thank you so very much for patient listening.
Thank you, Dr. Jaya. That was a very, very comprehensive lecture. And I think Dr. Aruna Verma is there. So Dr. Aruna ma'am, if you are there, you can summarize Dr. Jaya's talk.
Yes, ma'am. Yes, ma'am. Actually,
And
that time my microphone was not working, so I
no,
was
we are
not
so
able to.
glad Dr. Purva spoke so highly of you. So I really wanted you to chair Dr. Jaya's
It is
session.
not like this. I am very low profile
So,
person.
yeah,
ऐसा नहीं है.
thank you for joining. I hope you enjoyed the talk.
Thank you. Thank you. I have very much enjoyed Dr. Jaya's talk, and she spoke very well. So I think the main message from her talk is like whenever a pregnant woman is coming with anemia, we should screen for hemoglobinopathies also, especially the beta thalassemia, because it is common in our scenario, and we are not basically screening for that. If we will screen, and we can screen just by doing the simple hemoglobin levels, indices, as well as the general blood picture. And after that, we can just calculate the Menzel index just to differentiate between iron deficiency and thalassemia. So it is very important, and it is very easy to just screen the patients for thalassemia, and then we can go for next of a good screening test. And whenever we are getting it abnormal, we can go for confirmatory. It is not likely that we should go for confirmatory test at a stretch. So it is very important. And when we just diagnose it, we can just do the counseling and prevent the many complications going on in the fetus. So Dr. Jain explained each and every hemoglobinopathies very well: sickle cell anemia, Barr disease, alpha thalassemia. But in my view, the beta thalassemia is very common in our scenario. So we should screen. We should just can go for universal screening for beta thalassemia, just to prevent the many complications in the baby as well as later on. So thank you, Dr. Jay, and thank you, Dr. Monica. Just called me for this webinar, and I just enlightened by the talk of Dr. Jay. Thank you very much.
No, I mean, she is she is a doctor par excellence plus author. So we learn from her every time. I mean, it's so complicated that now
Wonderful
I have
talk.
to listen to
Wonderful
the
talk.
lecture again. One Sunday, I'll listen to the lecture again on YouTube. So, Dr. Jaya, I
Thank you so
wanted to
much, ma'am.
ask you, see, we are basically ART people, and our patients come for a workup before we start them on an IVF cycle or an IUI cycle. You tell me, they nowadays, like though it is not in the guidelines, we are getting HbA1c done on every patient because we
Absolutely.
just got random patients.
But
who
ma'am,
say,
actually,
"I'm
I
IVF."
am working in a government medical college.
So, madam,
Yeah.
so I was wanting to ask Jaya, madam, that madam, what are the cost-effective therapy, the investigations we can advise on the first visit for a couple planning pregnancy?
Actually speaking, we in India have too many Indias. We must first, acknowledge that there are different practices in different parts of the world, depending upon how logistically endowed we are. So
No,
if we talk of,
Dr. Jia, sorry to interrupt. We are talking about an India which is affording, which is
yes,
for IVF in a private facility, and which wants to be screened before planning
absolutely,
the conception.
absolutely, ma'am.
Yeah.
So to answer your query, there have been attempts. Now we have a Sickle Mukt Bharat kind of an abhiyan. We are talking of thalassemia-free country. So when we talk of all of these visions, every kind of identification of thalassemia or other disorders, depending upon which geographical area we are located in, has been attempted. But what happens is, ideally speaking, thalassemia or whatever hemoglobinopathy we are talking about is there in this girl even when she's going to a school. That time also she can be made aware of her status, right? Then ideally it should be pre-marital identification so that she doesn't actually get a partner who is going to be affected, or maybe a thal minor, and then they come together and give rise to a problem like this. Then it can be a preconceptional idea, like you said, when they're coming to you for an IUI thing. So all these are, natural course of the disease is such that we can identify a carrier in any of these parts. The problem that arose was Society of Fetal Medicine. We discussed this at length. This was a thalassemia day, kuch din pehle, it was there 22nd of May. So we were discussing in detail about this, that when we actually went to the thalassemia endowed belts and started doing school screening programs, what we ended up doing was girls started getting stigmatized. when they got identified as thal carriers. So one has to look at a bigger jigsaw, the social aspect or the implications of the screening program. Baki, as insofar as speaking on a scientific forum is concerned, for all the patients who come to me for a preconceptional counseling, HPLC is a mandatory thing that I do for them for both the partners. Especially if they are coming to me for preconception ki jagah early pregnancy visit. Because it is a time-bound exercise, I offer HPLC for both the partners because very often, until and unless the mother comes with a low CBC, and then I offer an HPLC, then I get a report, she is already gone past the 12 weeks window when I could offer a CVS. Because I have to also do a mutation testing thereafter. So in clinical practice, I come from a government college in Delhi, and we are offering HPLC to every pregnant woman and every preconceptional counseling patient.
Right. So HPLC, I mean, we had a lecture by Dr. Mandakini Pradhan last year, and we were so motivated that she also quizzed like you, and she's also a great teacher.
He is a master, ma'am. He's a teacher par
So
excellence.
this time, I mean, I could not get hold of her. She was so busy, but you are just as good. I mean, you're excellent. So I made HPLC a part of my routine testing.
Absolutely.
Somehow it fizzled out, but now I'm again highly motivated because the population nowadays is so fluid. It's not the belts do not exist. Like international community in India, people are going all over across the borderlines of the states and staying everywhere. So you have pockets of Odisha in Mumbai, you have pockets of Maharashtrians in MP. So, I mean, it's always like this. So it's a good idea to screen. And as far as IVF and IUI is concerned, I have had a couple where the male was a carrier and he opted to choose a donor IUI, donor insemination for his wife. And some people can be so lucky. So thank you so much, Dr. Jaya. And we are waiting for your next talk.
Dr. Monica, can I just
yes,
ask
Kavita,
something?
please.
I'm so sorry I'm interrupting. I'm not a
No,
chairperson.
no, please go ahead.
No, ma'am.
Yeah.
So actually, I absolutely agree with Dr. Jaya when she says for all pregnancies, we have to do HPLC and also include it as a major recommended investigation for preconception counseling as well. But there was a slide where you mentioned where if they come as this thing comes as thalassemia major, then you have to offer them termination of pregnancy. So do you do that for all cases?
Ma'am, it depends. Like, there are conditions like Downs that we are screening for. Now, Down is a different
I'm
situation.
talking about thalassemia and
No,
hemoglobinopathies.
the reason I'm giving you this example is, ma'am, I think truth is very strange and much stranger than fiction. So when I started studying about sickle cell disorder and I went to these belts of Jharkhand, where sickle cell is very, very common, as you must be aware, what we realized was that there are women with sickle cell disease. They have partners who have sickle cell disease. And when we offer them prenatal testing, they get it done. But when the baby comes as sickle cell major, they say, हमारी जिंदगी भी तो चल रही है, तो उसकी भी चल जाएगी. We are not going to terminate. So, insofar as prenatal diagnosis is concerned, you and me are merely facilitators in a journey, and we have no business to determine whether the pregnancy will be terminated or not. That decision lies in the court of the parents only.
Yeah, this is what
That
I
is why I
wanted to
said
listen
down ka
from you.
example. This is
what I
ultimately,
wanted.
absolutely, ma'am. The patient is the
This
king,
is something to
and
do
we are
with
only here to
education
assist.
also. Dr. Jaya, this is something to do with education. Dr. Kavita, there are people who come, uptown in urban people, semi-urban and urban areas, and they get termination done for trisomy 21. They get termination done for polydactyly. Can you imagine?
Yes.
Choroid plexus cyst.
I mean,
So I think I come from a very different
it depends on
background.
the psyche of that
because-
patient. What she's going through and what that couple thinks. They might be overthinking couple or they might be simplistic like the
Yeah.
Jharkhand couple. So you have to give that, option to the patient and leave it to them.
So I think people in Punjab are more knowledgeable about this because I belong to Punjab and I'm working in a medical college here where we have a full fledged bone marrow transplant unit. It's working very well. So what we tell our patients whenever there's a possibility of major, we do advise them medical termination of pregnancy, but then we always give the option of bone marrow transplant as well. And I have seen couples personally in my practice in the medical college. Some of them really want to keep the pregnancy and go on for bone marrow transplant as well. So just wanted for the audience also to know that these two should be offered, and it is on the onus of the parents to decide what they want. Just wanted to bring that out. Otherwise, Dr. Jaya, it was beautiful, very nicely explained step by step.
Very
Thank you so much, ma'am. This particular aspect of it was very pertinent to this discussion, and I'm glad you brought this, ma'am. Thank
Yes.
you.
Thank you, Dr. Jaya.
Also brings a lot of regions into the discussion. I'm
Yeah.
talking from the central part. Nidhi is there from Sagar. Dr. Jaya is in elite place of Delhi, and Dr. Kavita from Punjab. So, I mean, we all of us can pool in our opinions because we are dealing with a particular
ma'am.
social
Region,
network, a culture, a community. And lots of Sindhis also, the Northwest people in coming to Punjab, you know, like I also belong to Punjab. So all my grandparents, everybody was from Lahore. So it depends in, you know, the type of patients that drain into your area. So Dr.
True that.
Nidhi, Dr. Nidhi has joined. Hi,
Yes.
Nidhi. Hi, Nidhi.
Yes, ma'am. Hello,
Yes.
ma'am.
Hi,
Good
hi.
evening,
So,
ma'am. It was
yeah.
lovely, Jaya ma'am, listening to you, and it was very enlightening. I was actually, see, I am speaking as a patient, not as a doctor, because I myself am Thal minor actually.
Oh, wow.
Beta Thal. And we got to know about it. I had a very difficult pregnancy. After 10 years of marriage, I had conceived, and my hemoglobin could not go above 8 even after 4 blood transfusions. So, and we got to know that it is Thal minor only 6 months after my delivery. So this was a very a topic very close to my mind, to my heart actually.
So
I'm glad you're a part of this, Dr. Nidhi, because you know that perspective mellows us down a lot. It tells us that what happens to real people, and we cannot take the patient away from the jigsaw, you know. We sometimes become very authoritative in our discussions, and we have to understand that at the end of it, it's their life.
It's actually, ma'am, I'm talking as a patient. That's why I'm saying what Dr. Kavita ma'am and Monica ma'am said. Ki, as I told you, mine was a very difficult conception and very difficult pregnancy. So, had I been diagnosed with Thal minor or major during the pregnancy, had the fetus been diagnosed, I would, being a doctor also, I don't think I would have ever opted to get it terminated actually. It would never have been an option with me, ma'am.
Times are
So,
changing. These days, people are not terminating down babies. Many of them come and ask us, get us the list of support systems that we would require. There was a patient, I remember, a Muslim couple who came to me with an autosomal recessive disorder, bored femur, typical radial ray defect. And they said, मैं ये पूछते नहीं आया हूँ कि pregnancy terminate करनी है, वो तो सबने मुझे बता दिया था. I have come to ask you, अगर ये bone सीधा हो सकता है. In utero, there is a therapy to do that because I am not here to terminate this pregnancy. I said ये बच्चा हो सकता है चल ना पाए, इसको हमेशा prosthesis की जरूरत पड़े, तो जिंदा तो रहेगा ना. I am willing to take it forward. I mean, hats off to the courage of these parents who start, you know, that they become parents in their minds long before they become parents physically. And that needs to be appreciated, and we need to be very mindful and sensitive in our counseling. Straight away, हर एक को ये बोलना तुम्हारा बच्चा ये है, इसको terminate कर दो, is a very brutal idea, very obsolete in today's clinical practice. That
Very bad.
empathy is extremely important that you're talking of somebody's child. Let's not forget that in any of our discussions.
I think all of you female medicine people are very, you know, gentle at heart. Dr. Jaya, even Ratna madam, we had her Ratna Dua
Definitely.
in our last webinar, and she was just talking on 14th of May, and she is all heart, you know.
True
When
that.
she counsels, when she's doing her, you know, counseling, teaching, it's like I don't know how I, you know, got her as a speaker. with a busy schedule, but she's like, I think because you see so many troubled couples and so many babies and feti like this, you become very gentle and very patient to the patients. So I request Dr. Shikha Gupta now to start session 3. Shikha, are you there?
Ma'am, we have introduced the chairpersons for session 3. Dr.
Okay.
Jaya ma'am can help.
Right,
So
right. So Dr. Jaya, you can again start the
I
talk.
can start with the NT thing now. So this time we're going to discuss a thing on NT scans. It's a very popular idea these days to offer a nuchal translucency scan between 11 to 13 plus 6 or 14 weeks. And we are here to discuss some of the nitty-gritties of what all can be offered in an NT scan. We call it the NT scan, but it has now become the first trimester anomaly scan. Now, I do not know how to get rid of this bar on the top. I think it must be bothering again.
Yeah, it's there in the upper 1/6 of the slide. So what
Yeah, and I'm not
Dr.
quite
Doyle
sure as
said,
to
Doyle said
how to
something
do away
on
with this.
minimizing.
I don't know what was done to
Actually, there are
keep this.
3 buttons. You have a Mac or you have a this thing?
Ma'am, I have an Android PC.
Android.
Can I minimize this somehow? Optimize, hide video panel.
She said something about minimizing the.
Yeah, I mean, I
You
seem to
the
be
Mac is in the
getting
upper
lost doing
left
it.
corner. In an Android, it is on the upper right corner. The 3.
Upper right, I have this show meeting, slide control, pause, share chat, participants. I mean, a moment ago I was able to do this. I don't know if
Support
I
team, can
can.
you help? Vishal, can you help?
Can you just suggest something to do this?
Support
Ma'am,
team.
actually, it is the Zoom error. We have
Support team can
update the Zoom application, ma'am.
Update
That
the
would
Zoom
take some
application.
Yes,
time. I
ma'am.
think
So
that
right now it will take a time.
You
Yes,
want
and besides, it
want
was
to
there the
send the
last time we
slides
could do it.
Dr. Jaya, you want to send the slides to the our studio? They will run the slides if you want.
No, that will be fine. Only I don't remember if there were too many what do you say, animations in it. So that becomes a problem.
Okay, then we'll
Like,
continue as it is. We'll continue as it
will
is.
it be okay? I'm not
Yeah,
too sure
we
as
are
to how
seeing
to
we are
get
seeing
rid of
maximum
this.
of the slide. No, we are seeing
I
maximum of the
I
slide.
think it's gone a little up. I don't know how,
Yeah,
but
it has.
yeah.
Yeah.
Oh, I'm so glad it happened on its own. I think it's the Zoom is listening to what we are saying, probably. So then I come to okay, the outline. So you'll be discussing when do we offer this scan? How do we do it? What are the aims? What that we want to achieve from this scanning? And we want to revisit some of the basics of ultrasound. And we have to understand that this is going to be an aneuploidy screening opportunity. We will also be screening for certain malformations, which can be screened for with certainty at this particular gestational age. We're going to look at what can we do for multiple pregnancies, whether we can screen for preeclampsia and FGR, as this is an opportunity to do a uterine artery PI. And finally, we will have a summary of whatever we can see. So first of all, when to offer. And this is a gestational age window of, as we say, 11 weeks to 13 plus 6 weeks. Now we have also come to the idea that we first measure the CRL and make sure that it is between 45 to 84 millimeters before we proceed further. And actually 84 millimeters corresponds to a little more than 13 weeks and 6 days. So it is always better to take the patient up for a scan, zoom to see the CRL. And once you have done that, then it is easier to proceed further. How do we do transabdominal or transvaginal? We do a transabdominal scan first, and if we want to look at a finer nitty-gritty of the human anatomy in case of the fetus, then we resort to transvaginal scanning. Sometimes morbid obesity of the patient precludes transabdominal views, and therefore we have to resort to a transvaginal because that approach gives us more proximity to the fetus that we're going to study. So the aim of the exam is to confirm dating first of all, because CRL gives you an idea of the exact dating. And if there's a discrepancy between the dates, the patient's dates and the fetus in question, then we can redate the pregnancy based on information obtained from this ultrasound. Number of feti, whether the pregnancy is single or multiple, can be seen. The chorionicity and amnionicity can be determined in case of multiple pregnancy, and that's going to be vital to the future course of action in these disorders, in these pregnancies. We can screen for fetal aneuploidy by looking at the fetal nuchal translucency, to examine the nose and the palate, to look at the fetal heart rate, to assess the blood flow across the tricuspid and the ductus venosus Dopplers, for early diagnosis of fetal anomalies, and as we said, for predicting risk for preeclampsia and fetal growth restriction. Coming to the basics, you have to always stick to a thermal index of less than 1 and a mechanical index of less than 1. These are indices for safety, and you must never exceed these. These are usually found in the top right corner of the screen for the ultrasound machine. And you must always bear in mind that you cannot exceed 1, because that adversely tends to affect the fetus in question. So the time has to be as low as is reasonably achievable. We call it the ALARA principle. The recordings have all to be taken during periods of fetal quiescence, during voluntary maternal breath hold, because the fetus is going to be moving all the time and you have to capture one frame, perfect mid-sagittal view of the baby, and then you can zoom various parts of it to picturize the nuchal translucency in one of them, DV in one of them, and of course tricuspid regurg will be taken in a different plane. Color flow mapping is not mandatory, though it is helpful in identification of the vessel of interest and in defining the direction of flow. So coming to this particular thing on this right, what you see here is that the angle of insonation has been explained. So when you see that angle of insonation, this is the beam of the ultrasound, and that is the flow of the vessel. When it is approaching in the same direction as the ultrasound beam, you get a flow like this. It's a positive wave. When you are perpendicular to the direction of flow, you get blunted waves like these. And when the flow is away from your transducer, the flow gets inverted. The Doppler, this thing gets inverted. And therefore, it is important to understand that the more the angle of insonation, the more you approach 90 degrees, you're going to get values which are erroneous. At all possible times, you have to insonate the vessel in such a manner that angle of insonation remains close to or less than 20 degrees. As close to 0 as is actually possible will be a great idea. Coming to nuchal translucency, it is a collection of fluid behind the fetal neck, can also envelop the entire fetus in more severe cases. The incidence of aneuploidy and syndromic association is related to the measurement of the translucency, and therefore measuring it is important. Why is the fetus to be taken at 11 to 13 plus 6 weeks? The reasons are manifold. First of all is 11 weeks is selected as the lower limit, because whenever you have to offer a confirmatory test because of the risk of aneuploidy, you cannot offer a CVS prior to 11 weeks. Secondly, this is an opportunity to look at various fetal malformations, and a very small baby will not be as amenable to study in terms of malformations, and therefore we do not offer the scan prior to 11 weeks. Having said that, these days pre-NIPS scans are also in vogue, and many malformations can be identified given the good resolution of our machines and better training of our personnel. 13 plus 6 weeks has been taken as the upper limit because the fetus gets more vertically aligned as it grows beyond the 14 weeks window, and therefore it is impossible to get the kind of perfect sag that we want. The second is that sometimes the collection behind the fetal neck, the nuchal translucency that we say, if it is more in the 11 to 13 plus 6 weeks window, then it tends to get resolved in the second trimester, and it is important to catch it when it is there. Because resolution later does not mean that the fetus is not suffering from any abnormality. And the third is that the sooner you identify the aneuploidy markers, better is to offer a first trimester termination just in case it is indicated, rather than go for a second trimester termination, which is not just emotionally, but also physically more taxing for the woman who is pregnant. So measurements for the nuchal translucency, the fetus head and the upper thorax have to occupy the entire screen, or at least 2/3 of the screen. A perfect mid-sagittal view has to be taken, where you are able to see the nasal bone and the rectangular palate. They should be clearly seen, distinctly seen, and the zygoma should not be visible on that particular plane. If the zygoma is visible, then you can falsely allude to it as a nasal bone, and you can miss an abnormally ossified bone, an unossified bone to be precise. The fetus has to be neutral in position. The measurements will be falsely increased if the baby is having a hyperextended neck, and they can be decreased with a fetal flexion position. And therefore, it is important to notice that there should be fluid between the chin and the thorax for the baby to be called in neutral position. Wait for the fetus to move so as to differentiate between the fetal skin and the amniotic membrane. The fetus has to jump for you to differentiate that you are not taking the lower limit of your nuchal translucency at the amniotic membrane. Amniotic membrane is not to be included. Translucency is totally within the baby's neck. The measurements have to be inner to inner, and the maximum part of the nuchal translucency is where you measure. So you cannot measure at any of the red parts despite that being nuchal translucency. You have to measure the widest portion, and your transverse cursor has to be in line with the inner portion of the translucency. For this to be measured, the gain has to be minimized so that contrast is enhanced and you are able to exactly identify the inner portion of the nuchal translucency. Taking more than one measurement is great, and report the maximum that meets all the criteria. Do not make the error of making an average out of those values. Why I'm discussing all this is you may not be able to do all your scans yourself, but it's very important in aneuploidy screening that you look at the images provided by the provider, because sometimes higher values of nuchal translucency get missed because the zoom is not appropriate, the fetus has not been properly imaged, and those images can give you an idea of whether that NT can actually qualify to be a part of the combined first trimester screen or not. Secondly, one must identify the causes of an increased nuchal. They can be cardiac defects or dysfunction. This can also be secondary to a superior mediastinal compression, as in cases of a narrowed chest in cases of skeletal dysplasias, or due to pathologies such as a diaphragmatic hernia. Altered composition of the extracellular matrix in conditions like achondrogenesis, achondroplasia, etc., can also give rise to a raised NT. Most common, the reason why NT is enlarged is due to ineffective lymphatic drainage, which can be a part of fetal echinacea deformation sequence, or also genetic syndromes such as the Noonan's syndrome. Fetal anemia, as in case of alpha thalassemia, Diamond-Blackfan anemia, or Fanconi's anemia, can also present as raised NT. Fetal hypoproteinemia secondary to congenital nephrotic syndrome can be an entity. And finally, conditions like parvovirus infection can also give rise to a raised NT. And therefore, raised NT is not only synonymous with Down or Turner syndrome alone. The implications are such that with a nuchal translucency more than 95th centile for the gestation, the more than 75 to 80% of babies with trisomy 21 are going to have this finding. Having said that, majority of cases, the nuchal translucency resolves and the babies are born healthy. This last line is extremely important because when one cannot offer a termination of pregnancy or scare the patients away to such an extent that they resort to a termination of pregnancy, irrespective of your opinion, because you have given them an idea about a raised nuchal. It is extremely important to emphasize that a raised nuchal means investigation. A raised nuchal does not mean termination of pregnancy. So it requires further workup. The first and the foremost step is to rule out aneuploidy, which will be done by a chorionic villus sampling. And what are you supposed to do with the chorionic villus sample? You're supposed to send it for a FISH or a QF-PCR first, which gives you an idea about the 5 most common chromosomes, that is 21, 18, 13, and the sex chromosomes. And if that turns out to be fine, then you have to go for a microarray, which is a higher test, and we normally offer a medium resolution, that is 750K microarray for a raised nuchal. A detailed mid-trimester evaluation for malformations must be offered, and fetal echocardiography, both in the first and also in the second trimester, if the first one does not show up any major abnormalities, is mandatory. A genetic evaluation for alpha thalassemia should be a part of the workup if the mother has a microcytic blood picture also. Please do not forget to reassure the couple that even with a raised NT, the baby can be absolutely normal. What if the increased nuchal does not resolve in the second trimester? One must test the mother for congenital infections such as toxoplasmosis, CMV, and parvovirus B19. Follow-up scans should be offered every 4 weeks to define the evolution of edema. One must offer DNA testing for conditions like Noonan. We had patients wherein the QFPCR and the karyotype, everything turned out to be normal, but the nuchal did not resolve in the second trimester. And because DNA had been stored on the CVS sample, we offered further testing, and the baby actually turned out to be a Noonan's. So it is important to offer DNA storage whenever you're offering prenatal sampling, whether it is CVS or amniocentesis, because that DNA storage can facilitate further testing right up to exome sequencing and whole genome sequencing should the need arise and if the patients are willing. One must counsel that even when the nuchal is not getting resolved in the second trimester, there's a 10% risk of perinatal death and the live birth rate with a genetic syndrome that cannot be diagnosed prenatal. Risk of neurodevelopmental delay in such cases is going to be between 3 to 5%, and this should be told to the prospective parents. Coming to fetal heart rate, a 4-chamber view of the heart is obtained, and a pulse wave Doppler is used to obtain about 6 to 10 cardiac cycles, and the FHR is calculated by the software. One must remember that all trisomy 21 patients, there is a certain association with gestational age. This is the heart rate on this side and the gestational age on this side. With increasing gestational age, I'm talking of the number of days, the heart rate actually tends to fall. For trisomy 18, in euploid fetuses, at around 5 weeks, the heart rate is actually low in a normal baby. Then it tends to rise as the fetus moves to the 10 to 11 weeks window, and it comes down at 14 weeks. In T21, FHR is mildly increased to more than 95th centile in about 15% of cases. Trisomy 18, it is mildly reduced. But in trisomy 13, it is extremely high in 85% of cases. Remember that both Pape and beta HCG are low in trisomy 18 as well as 13, and fetal heart rate becomes a marker to distinguish between the two. Trisomy 18, it's going to be lower side. Trisomy 13, it is going to be higher side. Coming to the nasal bone, again the same window that you had chosen for the measurement of nuchal translucency is going to be utilized. You will see an absolute mid-sagittal view. There will be an equal to sign where the lower part of the equal to is the nasal bone, the upper is the overlying skin, and here is the tip of the nose. All the three have to be identified, and zygoma should not be visible. The rectangular palette should be visible for you to be able to say that this is the right plane for nuchal translucency. In addition, it is important to see that the bone should be more hyperechoic as compared to the overlying skin. If it is the same echogenicity as the skin or less than the skin, then this will be an unossified nasal bone. For this to be identified properly, the gain of the machine has to be reduced after focusing so that you are able to identify the echogenicity clearly. And even if the nasal bone is unossified between 11 and 14 weeks at the time of your scanning, you do not again have to scare the patient. You have to just ask her to come back at 16 weeks and look at this nasal bone again. If it is now ossified, nothing needs to be done. Nasal bone is absent in 2% of euploid fetuses also. 60 to 70% of Down syndrome babies are going to have an absent nasal bone, and around 50% of babies with trisomies 18 and 13 are going to have absent nasal bone. However, if you are looking at the baby too soon, at around 11 weeks, you are likely to identify an absent nasal bone. Of course, it is going to be more prevalent with instances of raised nuchal, and also when the mother has a Black ethnicity. Absent nasal bone between 11 to 14 weeks is not an indication for termination of pregnancy. Please remember this. Coming to DV. DV is a small vessel that is about 2 millimeters in size. So you have to identify this by zooming in the portion which includes thorax and the abdomen. And you will see that this is the umbilical vessel coming in from here, and this is the inferior vena cava. The connection between the inferior vena cava and the portal system this side is going to be aliasing a little at orange. And this is what is your DV, and you are supposed to focus it by putting your gain at again about 0.5 to 1 millimeter, because it's a narrow vessel. If you have too much of gain, then you will be getting contributions from other vessels also in the vicinity, and that will hamper your deciphering the DV Dopplers appropriately. The filter should be set at a low frequency, and the sweep speed should be kept higher, 2 to 3 centimeters per second, to be able to do this. Of course, the fetus should not be moving at this time. There are three waveforms here: the S wave, the D wave, and the A wave. Every venous waveform has these three patterns, S, D, and A waves. And it is the A wave that we have to look at, also the wave for atrial systole, as we call it. It has to be positive. Even if it is absent, it is fine. It should not be reversed. If it is reversed, that is a marker for aneuploidy. Qualitative assessment of ductus venosus blood flow is based on the appearance of this A wave. A reversed A wave is associated with, of course, chromosomal abnormalities, but also cardiac defects and also the risk of fetal death. These days, ductus venosus is an entire saga in its own right, because it gives you a reflection of the status of cardiac function in the baby. But I will not be going into the details of that. Suffice to say that in about 80% of cases with reversed A wave, the pregnancy outcome is going to be absolutely normal. And therefore, once aneuploidy is ruled out, a fetal echo should be mandatorily offered for ruling out cardiac malformations. Again, a reversed A wave between 11 to 14 weeks is not an indication for termination of pregnancy. The risk of miscarriage or fetal death between 11 weeks and delivery is about 2%. And therefore, if a DV is A wave reversed, one should monitor for fetal growth with scans at 20, 28, and 34 weeks, and also monitor uterine artery PI and fetal Dopplers. Coming to tricuspid regurg, you see a 4-chamber view of the heart here, and you have to place your cursor, of course, on the right side in the region of the tricuspid valve. Remember, there are three valves in the three leaflets in the tricuspid, and therefore you have to place your cursor at 1, 2, 3 positions to be able to interrogate separately each leaflet. And color flow mapping should not be used, as it is unreliable for the diagnosis of tricuspid regurg in the first trimester. Remember that sample grid should be large in order to interrogate the leaflet appropriately, between 2 to 3 millimeters. The sweep speed should again be high. Regurgitation lasting during approximately half of the systole and with a velocity of more than 60 centimeters per second is a cause of concern. So this is a normal waveform, and this is an abnormal waveform. You see that this is going down to more than 120, and we say more than 60 is what is the cutoff to say whether it is normal or abnormal. Moreover, it should be present for at least half of the systole. So this is more than half of the systole, and that's why it is abnormal. Coming to the risk of cardiac malformations, increased NT is associated with increased risk of cardiac malformations. A reversed A wave on DV Dopplers increases this risk further. Positive or absent A wave reduces this risk. Tricuspid regurg increases the risk, and absence of tricuspid regurg is protective for cardiac malformations. So when we come to the screening for trisomy 21, we understand that using only maternal age will only give us 30% of women who are going to carry children with Down syndrome. We can improve this by using a combined test, which means a dual marker with fetal NT with maternal age. We can also add more markers like the nasal bone, ductus venosus, or tricuspid flow to improve the detection rate to 93 to 96% and reduce the false positive rate to 2.5%. Quadruple marker is a second best choice. So whenever given an opportunity, you should always recommend combined first trimester screening between 11, 13 weeks and 6 days. Coming to NT scans, these are also an opportunity to look at fetal malformations. So here you see that the fetal central nervous system can be called normal when you have this concept of spaces. You can see that a small brain in the initial part that you see has this butterfly appearance, which is essentially ruling out holoprosencephaly. When you see this midline bar, we call it the fox. When it is present and you have these two butterfly-shaped choroid plexuses occupying the brain, then you can say that this brain is more or less okay at this point of time. Then you can find the concept of spaces. We have three spaces. We have the brainstem in the upper part. Then we have if you go back, this is the nuchal. This is the choroid plexus of the fourth ventricle. You have the cisterna magna behind it. And this is what is the intracranial translucency. Intracranial translucency is again an important concept and should be measured in all of these scans that we perform to be able to identify disorders of the neural tube. Coming to the various sections here, I'll be taking them one after the other. So then we examine for the fetal spine. We're able to see the fetal ears also. This is the bladder. This is the stomach bubble in an axial section. The fetal heart, we have to have a 4-chamber view. We can also do a first trimester fetal echo because then we are able to see the 3-vessel trachea view. These are the outflow tracts, the right and the left. And we have the DV that we are supposed to see any which way for the aneuploid. Multiple abnormalities, like we can have a patient like this, a 32-year-old lady, a second gravida, at 12 weeks and 2 days gestation with a corresponding gestational CRL corresponding to 11 weeks. So that CRL is actually small. We can see fetal acrania and the fetal brain is surrounded by amniotic fluid directly. There's a large omphalocele along the base of the cord insertion. One hand shows a claw hand or the ectrodactyly. Bilateral feet are rocker bottom and the fetal heart could not be evaluated completely, but the DV shows reversal of AV. So you see here that the CRL is smaller than what is expected at the period of gestation. You're able to see the upper limbs. You can see the umbilical cord, the fetal spine. And you're able to see that the brain is surrounded not by a cranium. The brain tissue is hanging into the amniotic fluid. And then you see this omphalocele here. That's an omphalocele coming out at the base of the umbilical cord. And you're able to see that on termination, this turned out to be an anencephaly with an omphalocele. And you can see the abnormal hands. So all of this is possible in the first trimester. Now there's a list of what can always be picked up on a first trimester scan. So acrania, encephalocele, lobar holoprosencephaly, tricuspid atresia, pulmonary atresia, pentology of Cantrell, body stalk abnormality, gastrochiasis, ectopia cordis can be picked up with certainty in the first trimester scan itself. And therefore, it is good to guide your patients to get their first trimester scans done with centers which are offering an evaluation for fetal anatomy also. Coming to various kind of malformations that can be picked up, you see here the typical bars separating the right from the left is not present, and therefore this is an alobar kind of holoprosencephaly. Here the cranium is missing. You can see the frog eyes, and this is anencephaly. This is a persistent flexion of the fetal back, and this is what is called enencephaly. Here your CRL will always be less than the period of gestation, and this extension of the fetal neck is also fixed. No matter how long you scan, you will never see this neck getting straightened. And this is again not compatible with life and therefore should be picked up well in time. You can see the same here. This is an omphalocele. Early onset omphaloceles picked up in the first trimester tend to have poorer prognosis as compared to those picked up in the second and late third trimesters, and therefore should be evaluated for, of course, cardiac abnormalities associated with them and also for aneuploidies that are associated with them. This is gastrochiasis. So you can see that there is no covering of amniotic membrane of peritoneal membrane on the top. And you see that the gut is hanging loose into the amniotic fluid. This can be picked up very well in the first trimester scan. These are limb reduction defects. You can see this is a megacystis, which is a soft marker, and this should be evaluated further because it is associated with an increased risk of trisomy 18 and also trisomy 21. Here you see a body stalk abnormality where the cord is abnormally short. So these are examples of first trimester malformations that can be picked up. CNS, as we have already discussed, acrania, lobar holoprosencephaly, and encephaloceles can be picked up. CVS, AVST, DORV, etc. can be picked up. Abdominal wall defects such as exomphalos, body stalk abnormality, gastrochiasis, etc. can be picked up. Insofar as multiple pregnancies are concerned, it is extremely important to determine the chorionicity and amnionicity at this particular gestation because later on, chorion and levy are going to fuse, and beyond 20 weeks, you will not be able to certainly identify chorionicity, and therefore this opportunity should not be missed. You should always insist upon getting an image for the lambda or the inverted T sign whenever a patient is being reported with twin pregnancy. A twin pregnancy is not a diagnosis in today's times. There have to be adjectives like monochorionic or diamniotic, dichorionic. Unless you have a mention of the chorionicity and also of amnionicity, the diagnosis of twins is not complete. Of course, for traditional. Hello. I'm sorry.
No, ma'am, there is a disturbance. Support team, please mute yourself. Madam, please continue.
Thank you. So also a differential with the nuchal translucency of more than 20% is associated with a risk
Support
of
team, please mute
PTTS in the
everybody.
subsequent pregnancy, in the subsequent gestation, and therefore it should be viewed with concern. So discordance of CRL has to be reported, discordance of nuchal translucency has to be reported, and remember, for dating of a multiple pregnancy, the larger CRL is taken into consideration always. Diagnosis of trap sequence is possible in the first trimester and should be done with certainty. Also, the risk of aneuploidy has to be marked. Chronicity and amnionicity, we all know the twin peak or the lambda sign, or the inverted T sign, which is associated with monochorionic pregnancies, and entangled umbilical cords in a momo gestation. Monochorionic monoamniotic can also be picked up in this particular period of gestation. Coming to screening for preeclampsia or pregnancy-induced raise in blood pressure is a severe condition and it is a life-threatening condition in many instances. And screening is possible using the NT scan as an opportunity, and it can potentially improve the pregnancy outcome by surveillance offered, and also by giving us the opportunity to initiate low-dose. So one must always say that preeclampsia screening is a multidisciplinary thing. It is not just mean uterine artery PI, it is not just PLGF, it is also maternal high risk factors. And so therefore, whenever you're seeing a patient for the first time in her visit, take consideration of her age, ethnicity, parity, smoking, medical and obstetric history, and the method of conception, because all of these have a bearing on the risk of preeclampsia. Her metabolic risk can be assessed with a history of diabetes. A raised BMI is associated with an increased risk of preeclampsia. Also, there can be placental risk factors in terms of velamentous cord insertion.
One minute.
The contribution from biomarkers like
Vishal, please call the somebody is talking at their end. Please
Support
call.
team, please mute everybody.
No, they are only not listening, Vishal. It is the support team which is talking. So I think you will have to make a phone call.
So, and then comes the role of uterine artery Dopplers. Of course, the cardiovascular risk of the patient has to be assessed, and existing cardiovascular conditions, including the mean arterial blood pressure, have to be recorded. And then all of this needs to be put together to be able to assess the risk of preeclampsia. Why is the uterine artery the area of interest in this regard? It is because of the wave of trophoblastic invasion that occurs in a normal pregnancy. You see this media is getting lost. This was not getting lost in the case of preeclampsia, and therefore the vessels remain sensitive to the vasopressor influences of the hormones, and that is what gives rise to the condition of preeclampsia, and therefore uterine artery is what we want to look at. So measurement of impedance to flow in the uterine arteries renders this change quantifiable. Why should we use the PI? These days we see that PI is the ratio to be used, but we also used to use ST ratios in the years of yore. And why PI is to be used is, if you look at this waveform, the systole, the diastole, and the ratio between the two, and this dotted line represents the mean. So we have an ST ratio, we have a resistance index in the form of S minus D upon S, and we have a pulsatility index by the meaning of the difference between the systole and diastole divided by the average velocity of the cardiac cycle. The reason why we have chosen PI is that it never goes to infinity. It will always give a value, even if it is AEDF or whatever. So that is why it is important. One must, as we said, insonate always with an angle of insonation of less than 30 degrees. Measurements are to be taken at the point where the iliac vessels are being crossed by the uterine vessels, and these happen to be around the internal os at the time of the 11 to 13 plus 6 week scan. So you have to hold your probe vertically in the suprapubic region in the center and tilt the probe a little to identify the uterine vessels on both the sides. We can also use the transvaginal technique. However, most of our cutoffs have been calculated based on the transabdominal techniques, and therefore, so long as it is visible clearly and nicely, we should be using transabdominal pins. Coming to first semester PI, it is good to identify and remember these three cutoffs, 2.7, 2.5, and 2.38. However, normally we say that anything above 2.3 is abnormal for a mean uterine artery PI. Remember, placentation is not important. And therefore, even if it is a right-sided placenta or a left-sided placenta, you have to only take the average of both uterine artery PIs. And that, if it is more than 2.3, should be viewed with concern, and you should put it into a software to be able to see if the patient has a risk of preeclampsia of more than 1:100. And if that is the case, she becomes a candidate to initiate low-dose aspirin. With this, we come to the end of the session. The take-home messages are: scan around 12 to 13 weeks to minimize repeat evaluations. A TVS gives more information. So whenever you are in doubt, especially regarding amnionicity of a multiple pregnancy, if you're not able to see the amnion, please do not say it is a monoamniotic pregnancy until you have evaluated transvaginally, in which case you will be able to see majority of amnions. It is going to be of grave concern to the mother, because we'll have to terminate a monoamniotic pregnancy sooner than the diamniotic one. And therefore, please take pains to identify the membrane and see if a transvaginal scan helps you. Although an increased fetal NT is associated with abnormalities and fetal death, the majority of babies survive and develop normally. So one should use this opportunity to reassure rather than scare. And after the diagnosis of a raised NT, the aim must be to distinguish as accurately as and as quickly as is possible between those who are likely to have problems and those who are likely to be normal. Evaluation for malformations and screening for preeclampsia are an important part of the exam, and every opportunity should be utilized. And therefore, while you are writing on the patient's paper, OPD paper, a requisition for an NT scan, please do not fail to mention that you're also looking for a first trimester malformation or anomaly scan, and you're looking for preeclampsia screening. Once you have jotted this down on your paper, the radiologist is bound, radiologist or a fetal medicine person, whoever is performing the scan, is bound to give you that kind of information. So it is important to ask for it first. Lastly, the aim of the examination is to provide insights and not scare the patients away. Thank you so very much for a very patient listening.
Thank you, Jaya ma'am. You were really, patient with us, taking two such elaborate talks. Madam, just a quick question. Suppose our preeclampsia screening is fine during the NT-NB scan. We are doing a combined test. We are sending the dual markers. So should we send a placental growth factor along with the dual marker?
Ideally speaking, I do not have that talk on this particular system. Otherwise, I would have given you an example. That Fetal Medicine Foundation is offering us softwares which give us an answer of the risk of preeclampsia with maternal demographic factors alone. When you add a mean uterine artery PI and mean arterial pressure to it, whether you add a PAPA mom to it from the dual marker report, whether you add a PLGF mom to it with the dual marker report, in addition to the dual marker report, or if you're doing a first trimester quad or a penta. So what I'm trying to suggest is that whatever amount of information you have, there's a free software available on our phones to give us a risk of preeclampsia, and it can be utilized. If the patient is pocket friendly, he is able to afford PLGF, great. Even if the patient is not able to afford, every patient is giving us at least an age, a BMI, a mean arterial pressure. At least we know that the previous pregnancy was preeclampsia or not, whether it was lost to uteroplacental insufficiency. Sometimes we have inputs like APLA. Sometimes we have a chronic kidney disease or a chronic, hypertensive state. So there are a lot of inputs that can be gathered just by looking at the patient and taking a good history. And thereafter, whatever inputs we get will obviously refine our risk. So PLGF is very welcome, I would say. But I sit in a part of a world which truly is elite, and many of our patients are able to afford it. However, if it is not there, then this is not the reason to say that I cannot offer a preeclampsia screening because PLGF is not available, because we have a lot of things available. And I will take this opportunity to say that these days we are so busy looking at reports. We sometimes forget to look at the patients and talk to them. And a good history cannot be replaced by any amount of investigations that we offer as clinicians.
So, madam, you have given a very apt reply. And what is this app which we can download on our phones, all of us?
Ma'am, this is a fetal medicine. No, this is not even an app. It is a Fetal Medicine Foundation pre-eclampsia risk calculator.
Yeah.
They also have an aneuploidy risk calculator, because sometimes we get a dual marker separate and a NT report separate. And the person who is doing the combined first trimester screen forgets to put them together into a combined first trimester. So we have this online Fetal Medicine Foundation calculator where you put all the inputs and you get your risk assessment. So what I'm trying to suggest is that it is freely available. I wish I had this particular talk on the system. I could have given you that slide only. It is Fetal Medicine Foundation, aneuploidy risk calculator, pre-eclampsia risk calculator, stuff like that. So
Yeah,
the beauty of that calculator is that even if you do not have a PAPE or a PLGF, it will still give you a risk.
Right, right. And I think FMF has done some wonderful work to
Wonderful work.
educate all OBGYNs, and actually the website is also beautiful. The small videos they have put in the website are free to access for all. And the
Yes.
tests you can take too in order to improvise your NT scanning and everything.
I
So
think mean arterial pressure is something that we have underutilized as a subpopulation who has preeclampsia as a big problem. Between 3 to 10% of our population has preeclampsia, and merely using 2 digital sphygmomanometers and you doing this mean arterial pressure is something that can be easily accessible to all possible setups without any logistic constraints. And doing that can also go a long way in identifying. Moreover, dual marker ka PAPE, something less than 0.5 should always put a red flag there. कभी भी ये नहीं कि trisomy 21 का risk क्या है. One can always look at the moms. A beta HCG mom of more than 0.4, sorry, 4 moms. A PAPE of less than 0.4. Inhibin A of more than 2 moms. And the quadruple marker. All of these are biomarkers for our increased risk of preeclampsia. So it is raining information on the patient's file. Only we need to look at it from the perspective of a good clinician.
yes, so every report which comes in front of you, you should be able to, just see that figure 4.0, 0.4. I mean, this should be imprinted in your mind. Keep seeing the reports, keep, revising, reading, so that in your busy practice, you can really use this for the welfare of the patient. Thank you so much, Dr. Jaya Chawla. Words are not enough to thank you. We can only, express our love and gratitude for your great
Absolutely not.
time and a lot of time and patient teaching given to all of us. So
Thank you so very much.
any other comments, Kavita ma'am?
Yeah, Dr. Jaya, it was a superb talk, like the previous one. And I think you have really beautifully illustrated everything about the 11 to 14 week scan. You have talked about the technical and imaging details and also the various components of the 11 to 14 week scan. Many a times my residents in a medical college, when they are writing requests, I've seen them writing as NTNB scan. And the
Yes.
report also, it comes as nasal bone and nuchal translucency. They miss to see the ductus venosus and the TR and the other components. Malformations, obviously, they will be looking at, but also the uterine artery Doppler. So I think it is a good practice that we write all the details, because not being a fetal medicine specialist and we get our ultrasounds done by the ultrasonologist, we have to always re-emphasize what all has to be seen. I think that point has been really brought out very well in your presentation. And one more slide, I was really impressed by the fetal heart rate and the relation to trisomies.
13 and 18.
Yeah.
Yeah.
So
That's the
that was
only point
something
of
very
difference between
new to
these two.
me. And I'm very happy that you brought that out. And my question to you would be regarding that uterine artery Doppler once again. So if they report as right and left, we need to take out the mean and then see it on the software. Can
On
you
the
just
software,
elaborate
because
on that again?
as I said, ma'am, I mean uterine artery PI more than 2.3.
Yeah.
At the time of the 11 to 13 plus 6 week scan is what we take as a blanket red flag that this is going to be high risk. Number
2.3.
one, 2.3. So we all remember that value, even though at 11, 12, and 13 weeks, the cutoffs are slightly different. But since it is not important to remember them on a clinical basis very frequently, 2.3 is the upper limit that you put a red flag on, number one.
Yeah.
Number two, I think we, in clinical practice, of course, the software, Fetal Medicine Foundation, you put it as FMF a preeclampsia screening software or a calculator to be precise. It is a calculator and it comes onto your screen and you can just put in the values, whatever you know. They will ask you for the maternal age, they'll ask you for the BMI, whether the patient is hypertensive or diabetic or has had a preeclampsia in the past. They even ask you if the mother was preeclamptic. So if somebody is giving you that kind of information, even that can come in handy. If the patient is a chronic renal disorder or is APLA positive, SLE positive, all of these things would be asked. So if you're able to give them inputs in yes or no, even that can give you an idea of the risk of preeclampsia. Of course, a mean arterial pressure and a BMI can be taken at the very same time that you're seeing the patient. And then if you have a mean uterine artery PI, you can include that. If you have a PAPE value from the dual marker test, you can include that. And if you are, generous to have a PLGF mom also, that will be great. But what I'm trying to suggest in this entire presentation is that most of my target audience always are people who are not fetal medicine person per se. But we are raising a generation of obstetricians and gynecologists, all of us. So we have an onus on our shoulders to make them learn that they should at least look at the images of the report to be able to see whether the imaging is suboptimal or it is optimal. They should be able to gauge the quality of the scan because we are at the interface with the patient as a clinician. The person giving us that report is not responsible for the ultimate fate of the patient. But we
Right.
as OBGYN people are the people responsible. So to be able to say that this is gray, this is white, and to be able to spell out clearly, this is what I want. And if I am not getting it, I am asking for it again. Is very important because sometimes we see images jahan par baby is literally on a mountain and they're giving us an NT report. Now, this NT cannot be trusted. Even if we are saying that this is normal, I cannot trust it. Even if they say it is raised, I cannot scare the patient based on this image. So that is a window of opportunity. If the patient is less than 13 weeks and 6 days, we can always ask for a different scan from a different person, different place. And many a times we have revised our risk based on doing it ourselves again. So what I'm trying to suggest is that our residents, our clinicians should be trained to look at a scan and at least say, this is a good scan, this is a not so good scan.
Yes,
And of
and
course, the
as-
information provided.
Yes, and there was a radiologist. My neighbor is one of the famous radiologists of Bhopal. So she was telling me that, Monica, sometimes the scans which are coming from OBGYN, they come to me for repeat scans, and OBGYNs are very busy. They just put the probe and the fetus is hardly occupying not even 1/3 of the screen, and they've measured NT and told her
Absolutely.
it's normal and send a dual marker. So, I mean, the patient gets false reassurance, and these, when they meet somebody else, they send them for a repeat scan. So the patient gets the impression कि उसने ठीक नहीं किया, इसने ठीक किया. So I think there should be a uniformity. If you are doing a first trimester screening or scan, follow the simple guidelines and give adequate time for that scan.
Yeah. I mean, you can't afford to miss an anencephaly saying I was doing an NT and B scan. So that's not fair. I mean, you have to look at the baby in toto and give your report.
Yes.
Very important.
So
And now obstetricians are recommending in the 11 to 13 week scan to look at the cervical length and internal loss also.
And that is a part of first
Yeah.
trimester screening.
So
Ma'am,
that
it's not-
also that
it's
is
a very
also a
much part.
component. Yeah.
And measuring the mean uterine artery PI, measuring the cervical length, seeing
Cervical
the
length, internal
placenta,
loss, yeah.
seeing the TR, seeing the DV
Yeah.
is an integral part of first trimester.
Yeah.
screening.
And then to repeat the cervical length again to predict preterm labor
Yes.
when
Yes,
you do the
16
targeted
weeks is
scan.
when we
Yeah.
normally do that. And if the patient is at a high risk of preterm labor, then the ISO guidelines for on the subject say that 16 weeks onwards, we should be starting off our surveillance and accordingly starting out micronized vaginal progesterone just in case the patient merits that. So
Because
yes,
we talked about the combined test and the integrated test also is also there, but I was reading some papers and I think there was some presentation in a conference where we talk about cell-free DNA in the first trimester itself, along with the NTNB. So what are your points?
it's a very straightforward take home, ma'am. Since we've discussed that an NT scan is giving us a plethora of informations besides aneuploidy also. Number one, until we have confirmed the number of the fetuses, until we have confirmed that the nuchal translucency is essentially normal, we cannot request an NIPS. Because a raised nuchal cannot be addressed by an NIPS. Anything more than 99th centile, you have to put in a needle to be able to say
Yeah,
whether the baby is okay or not.
yeah. It's
So
not a diagnostic.
yes, the practice of saying NIPS over everything else is not okay.
Yeah.
You have to do a scan to at least confirm the dating first. Because if it is less than 10 weeks, you can't technically offer an NIPS prior to 10 weeks. So you have to wait and say at the 11 to 14 week scan that the baby does not have a gross congenital malformation, because that precludes the use of NIPS. You cannot offer an NIPS to somebody with an omphalocele maybe. So what I'm trying to suggest is that that information is mandatory for us to be able to decide whether we want a combined first trimester or we want an NT scan followed by a NIPS. That can definitely be a strategy, but NIPS cannot replace the scan.
And ma'am,
For
Kavita,
sure.
Yeah,
that is
definitely.
probably because of the whole lot of pressure of labs. I think every day you are getting one new lab who is coming to visit you. That madam,
For
the
an
pressure
NIPS.
is so much. They'll come out with packages and NIPS. Every obstetrician feels NIPS kara lete hain, now it's come down to this, kara lete hain, ye kar dete hain. But nothing can replace that initial steps which we take. And you should always
Scan is
NIPS for
mostly
initial
mandatory
screening.
always.
Yeah.
The scan is mandatory, but this particular paper had spoken about cell-free DNA replacing the first trimester dual marker test.
That is true. That is true, ma'am. But again, see, NIPS will not give you PAPE.
Yeah.
And the problem in our country, if you look at the percentages of preeclampsia in our mothers
Yeah.
and the problem of Down syndrome, then what is the percentage for Down syndrome? 1 in 700 to 1 in 800. Don't we
moreover,
see more
NIPT is
preeclampsia in our
expensive.
practice? So,
is
see, we have to take
costly
first
also.
things first, because Nicolaides is saying Down syndrome is something important. We cannot forget which part of the world are we sitting in. So our mother's gestational diabetes is a bigger problem. Preeclampsia is a bigger problem. Not knowing the amnionicity and chorionicity is a bigger problem. So we have to customize solutions to our problems. We cannot seek answers to our problems with the research from the West. Let's be very clear on that front first.
No, we are
We are a
just discussing it, whether in
doesn't
the
matter,
near
I
future
think.
it will
It should be
replace or
should
not.
be a well-documented meta-analysis. When it is totally proven, it will automatically come in the guidelines.
And American College, any which way says that NIPS as a screening for aneuploidy should be offered to every woman. Like
Yeah.
it should be told that there is a modality like that. And I do practice that in my clinical practice. I've seen patients coming with combined first trimester low risk, but the lady is 38 years of age. So I'm not getting convinced that she is low risk, because merely by her age she becomes high risk. So how can you combine first trimester prove it to be low risk? And then she turned out to be down.
Oh, by NIPS.
She was down by NIPS, then we put in a needle to confirm it. And of course, she was confirmed on that also. And then we had to go ahead and terminate that pregnancy. So it is extremely important. Patients, I mean, combined first trimester reports writing mode of conception unknown.
Oh,
Now, come
God.
to think of it, no matter how illiterate she definitely knows how she conceived, whether it is IVF or a spontaneous conception. ये तो वो lady बताएगी ना आपको.
Jaya
So
ma'am,
if you are
what is your take on NIPS in multiple gestation? Twins?
Ma'am, up to twins, it is validated. It can
Okay.
very well be done.
Right.
Beyond twins, you have to only look at NT and the maternal age for sure.
Yeah.
NIPT is related to maternal weight as well.
Yes,
The
but there is a
sensitivity.
component for it. NIPS, when they're asking you the details, they're asking you for the maternal weight and BMI. So again, the smoking status of the patient, the diabetes status of the patient, ethnicity of the patient. If it is writing on top unknown for everything, at least you know the ethnicity of your patient. Why do you say unknown for it? You do not know whether your patient is smoking or not. Is it not funny? You're saying twins unknown. I mean, which part of the world do you come from if you write twins unknown and perform an NIPS or a combined first trimester? So what I'm
madam,
telling you is that these
yeah.
reports will tell you where is it shoddy. It's written right there on the top. We forget to examine that bracket. We straight away go down to the report niche. But
Yeah.
I take a cue from that information as to how serious this lab is about providing a good report.
True. So we have a pro forma. Whenever his patient is coming for first trimester screening, this history has to be taken by the junior doctor. She gives that information to me. I make that report nicely so that nothing is missed. I mean, that is a protocol we follow. Of course, we have low volume of work, so we don't ask that, not stressed about time and everything. But this is mandatory if you want a good reporting.
Well, sometimes I think we cannot go down under pressure and say numbers is why our quality of work is coming down.
Yes.
No matter every teeming millions, every patient is individual and we are
Every
answerable to them if we are seeing them. We can turn down saying we cannot see you, but we cannot see them half-heartedly and say, I was under a lot of pressure, so I did not give you measure your BP on the antenatal visit. I mean, that is something unpardonable. No matter rich or poor, they have a right to quality medical this
So
thing.
people in
And
government setups with high
we can't,
pressure. Yeah. So,
ma'am, we always have a solution to a problem.
Madam, postgraduates in your hospital, are they doing scanning? First years and second year residents? Jaya, madam?
For us, it is actually the second years and third years. They are doing peripheral posting in ultrasound and radiology and other departments as well, like radiotherapy and all.
But
But
not
as
in
such,
the department.
routinely, we are not allowing them to do the ultrasound because we have to give a report and the आजकल बहुत hassles है. Patients कहते हैं आपके junior doctors हमें देख रहे हैं, ultrasound कर रहे हैं. And the PG curriculum which comes from the university is 2 weeks posting in ultrasonology. Otherwise, in
We, on
labor
the
room,
other
they are
hand, ma'am,
using it.
in
for
labor
fetal
room, ma'am,
heart
we have
rate,
a free
the central
hand.
localization. and-
So labor room 24/7, the machine is available, number one.
Yeah.
Number two, in order to do their biophysical scoring, I encourage them कि labor room में biophysical कर रहे हो, at least biometry तो कर लो. Baby का weight तो आ जाएगा. And by the time that they start doing the weight and the baby is coming out any which way in the next 2 or 3 days, so they're able to verify whether their weight was correct or not. So that gives them an insight, and then I encourage them with their thesis topics. One of my topics was maternal ophthalmic artery Dopplers, which I wanted to include as a part of the 11 to 13 plus 6 week scan, but I could not for this was an old presentation when I was not doing ophthalmic artery Dopplers. So now we are including that as a part of the first trimester scan because that gives me an insight into the preeclampsia risk again. So, because there's a thesis on it, the PG learns to do an ophthalmic artery scan, and trust me, it is easier than any obstetric scan whatsoever. आँख का eyeball तो कहीं नहीं जाएगा, तो ophthalmic artery भी कहीं नहीं डांस कर रही है, पूरा तो बच्चा ही डांस कर रहा होता है. So we are struggling with movements of the baby most of the times. Ophthalmic artery is one of the easiest things to do. Then one of my PGs did a pulmonary artery Doppler. She was trying to find out the risk of respiratory distress syndrome in preterm babies. So imagine a first year PG learned to do a pulmonary artery Doppler. It is we who are thinking that there is a limitation. It is in our minds. Our PGs are very sharp people. जिसको OBGYN का PG मिल गया है मैम इंडिया में, उसके IQ क्वेश्चनेबल नहीं है. हमारी डेडिकेशन क्वेश्चनेबल है. हम उसको सिखाना क्या चाहते हैं. They are able to learn everything and do everything. I'm telling you from my personal experience.
What are you using the pulmonary artery Doppler for, madam?
Ma'am, this was a thesis that we did on pulmonary artery Dopplers in the preterm fetus to be able to identify the risk of respiratory distress syndrome.
Okay, got it.
And it turned out that this was a valuable parameter.
Wow.
This could tell us which baby is going to land up into respiratory distress.
So
This was
Doppler
one of my
has
first PGs
Doppler
4
has
years ago.
many opportunities. So I think, ma'am, I think we have had a wonderful interactive session. And
Yeah.
Shika, would you just give us the vote of thanks, please?
Yes, ma'am. Good evening, everybody. So it's my privilege to propose a vote of thanks on behalf of the whole team Synergy. I would like to thank all the dignitaries present here. Our chief guest, Dr. Parul Purvala, sir. Our guest of honor, Dr. Ashok Kumar, sir, and Dr. Sarita Bhalerao, ma'am. And our special guest, Dr. Kanti Bansal, ma'am, to be present here and chairing the session. I extend a hearty thanks to all our eminent speakers, Dr. Doyle Bose, ma'am, and Dr. Jaya Chawla, ma'am, who enlightened the session with their thorough knowledge and their deep insight to the respective topics and delivering a very heart-to-heart talk. My special thanks to Jaya ma'am for helping us to continue this session and through her exhaustive back-to-back 2 talks.
Thank you so very much for having me here. Thank you.
My special thank to all the chairpersons who accepted to be a part of this session and making it more eventful by gracing the session with their presence and their kind words. I would like to thank both the program conveners and all the program coordinators for executing this program so nicely and for the flawless flow of events. I would like to thank Team Intas for working day and night to make this series a great success. And last but not least, I would like to thank all of you for imparting their valuable time from their busy schedule and joining this session. Now, I would have a privilege to invite everybody for our upcoming Synergy 16 series, which is following this June on multifetal gestation. Hope everybody will join to make this also a great success.
So
Now, I would
thank
like
you.
to
Thank you
finally
so
hand
much.
over.
Thank you, Dr. Vishal, and thank you, Shikha. And our next Synergy 16 is on multifetal gestation. It's called Double Trouble. And we are having it on 12th of June. And we'll see you again with some fantastic talks and some fantastic speakers. So long, Jaya ma'am, till we meet again. I'll be troubling you again. So long, Kavita ma'am. See you.
Yeah.
And bye, Nidhi. Thank you, Vishal. Bye,
Thank you.
Shweta. Bye, Shweta.
Thank you.
Thank you
Thank
so
you,
much.
Dr. Monica, and special thanks to Dr. Purva. When she called me up, she said genetics pe hai webinar. So I was a little skeptical about it, but I'm so thankful that I'm glad that I said, okay, I will be there. It was beautiful listening to all the speakers. Thank you, Dr. Monica.
Thank you so much, ma'am.
Thank you, Nidhi. Thank you.
Thank you, ma'am.
Have a great weekend. Bye. Bye.
Support team, please end the meeting.
Yes, sir.